Good morning, everybody. I'm Jaspreet Sidhu. I'm the Vegetable Crops Advisor with the University of California Cooperative Extension based here in Bakersfield, Kern County. And I welcome you all to our annual Carrot Research Symposium. And Maria has already gone over the housekeeping rules and the info on the credits. And with that, I would like to mention that we have a pretty packed agenda today and we have different topics to cover with two guest speakers. So without any further delays, I will begin with my presentation and providing updates on the Kern County trials that we did this year. Alrighty, so I'll begin with mentioning about the trials that we did this year. We did a couple of variety trials, then we did a cavity spot variety screening trial. We did the nematicide screening, we had pre-emergent herbicide screening trial, and an organic fungicide screening trial for alternaria leaf blight. And I'll go over these trials briefly about the results. So here is our organic variety trial, which was planted last year on August 7th, and it was harvested this year on January 10th. We had a field day where we had like good attendance and people evaluated these varieties in the field. And we had 10 entries in this trial, the reds from Organic Seed Alliance and from Dr. Phil Simon's USDA breeding program. And we assessed those 10 varieties for foliar disease severity, root weight, root shape, uniformity, and smoothness. And based on those criteria, there were a couple of varieties that stood out, like the ones I have shown here in pictures, 6220 and 6636. Those are the numbered ones. And with this trial, we also combined the domestication trial, where we had several entries from different parts of the world, and it was just for knowledge. And I will be compiling a report for the organic variety trial and the domestication trial, and will be sending out and sharing with the stakeholders in the next couple of weeks. Our next variety trial was the conventional variety trial, and we had about 102 varieties in this trial, 38 cut and peel, 34 cello varieties, and 30 colored varieties. And these varieties came from 5 seed companies and Dr. Phil Simon's USDA breeding program. And we did have a field day last year on June 23rd, where we had company people, people from industry and the stakeholders come out and evaluate these varieties in the field. Next one was our cavity spot variety screening trial, which is part of Dr. Phil Simon's breeding program. And in this trial, we had fixed 56 carrot lines, including the 5 cultivars, and also susceptible check atomic red. These were planted on 3-foot beds with 3 applications, and the trial was planted in October. We didn't use any fungicides in the trial, but just the regular herbicides. At harvest, the carrots were assessed for percent disease incidence, and disease severity index was calculated based on Dr. McDonald's work. And we also assessed the carrots and collected data on forking. And these are just the prelim assessments from the trial. We are still processing the data. And based on that, we had a moderate disease pressure in the nursery. The disease severity index ranged from 0 to 25% in the trial. The percent disease incidence varied from 0 to 47%, and the forking between 4 to 79%. Based on just the prelim assessments, we found that the trends were similar to Dr. McDonald's and Lindsay DeToit's work that they are doing with Dr. Phil Simon. And the susceptible check atomic red had the highest amount of disease in this trial. And we will be compiling and sharing the data soon from this trial as well. And these are just the visuals from the trial. You can see the cavity spot lesions on the carrots. And the right-hand side lower picture, you can see forking on different carrot varieties in this trial. Our next trial was the nematicide screening trial. And in this trial, we had 8 treatments. We had 2 Nimitz treatments, the Nimitz lower rate, 3.5 pints, and the Nimitz higher rate, 5 pints per acre. Nimitz is registered on carrots in California, but it has some issues with Fido and other packets. So we wanted to change a little bit of the approach here, because in the past we have seen reduction in plant stand and stunting, burning on the seedlings. So what we did was, like, Nimitz was applied 2 weeks before planting. It was applied on June 1st when the trial was planted on June 15th. So between those 2 weeks, we applied approximately 3 to 4 inches of water to move the product down from the topsoil profiles where the seeds are planted, just to see if it reduces the Fido. And we did see like little bit promising results. The Fido was reduced, and there was less I would say significantly less reduction in the plant stand compared to what we have seen in the past. So maybe moving forward, we will try to see if we apply more water or just play with some irrigation and then see if it reduces the phyto. It's a great product, but we just need to figure out how to use it. And then we had an organic option, MB1. We had one product from FMC, Slibro, which is registered in US, but not in California yet. And then we had a developmental product from Syngenta and then Vellum from Bayer, and it was used along with the soil surfactant Watermax. And at harvest, we assess the carrots for root galling, and this is the scale that we used. 0 to 3 are deemed marketable, and 4 through 10 are unmarketable. And this is just a visual guide that we use when we are assessing the carrots. And these are some pictures from our trial on right-hand side when we were taking the samples, and on left-hand side, carrots after washing, and you can see galls on the carrots. One thing we noticed was like sometimes the products don't move freely or quickly in the soil, and you can see on this left-hand side, like the top half of the carrots are perfectly clean, and then you see the nodes or the galls on the bottom. And this is the data on the root knot damage on carrot roots. Although all the treatments had less damage compared to the untreated control, but the 3 treatments, Vellum plus Watermax, developmental product from Syngenta, and Slibro, they had significantly less damage compared to the untreated control. The 2 Nimitz treatments, they were at par with each other, and they had moderate levels of damage in the plots. Next was our herbicide screening trial. We had 6 treatments in this trial. We had the untreated control, number 1, the hand-weeded check, and Lorox as a standard. Then we had dual magnum plus caprol, dual magnum plus caprol plus prowl, and caprol plus aptam. For dual magnum, we went with about 1/3 of the recommended rate because our trial site has low organic matter, and it's a sandy loam soil. For this one, the plots were 30-foot wide, and they were 30-inch wide and 30-foot long plots. And we took data for stand counts 5 weeks after treatments, and the treatments were applied a day after planting. This is a stand count from 1 square foot, and the hand-weeded check and caprol plus aptam, they had significantly higher stand count compared with the untreated control. The other treatments were at par with the caprol plus aptam and hand-weeded check, but they were not significantly different from the controls. These are our top weights per plot. The 2 treatments, caprol plus aptam and dual magnum plus caprol and Lorox, they had healthy looking green tops and really good canopies. And they had significantly higher top weights compared to the untreated control and the other dual magnum plus caprol plus prowl treatment. Similar to the last top weights, the yields were also higher in the 2 treatments, and they were significantly higher than the untreated controls in caprol plus aptam and dual magnum plus caprol. In caprol plus aptam, the yields were almost 50% higher than the untreated controls. And these are the visuals from the trial. As you can see on the top right-hand picture, caprol plus aptam, they had healthy looking tops and pretty good canopies compared to the number 1, which is the untreated control, and number 4, which is dual magnum plus caprol. And here on the bottom, this is just to show like we had a heavy extensive weed pressure in the untreated controls compared to number 6, the hand-weeded check number 2, and the number 4s. With that, for our plans for moving forward next year, we will continue to maintain the cavity spot nursery and continue screening biologicals and the variety screening trial for Phil salmon. And we will also keep expanding and maintaining the root knot nematode nursery to evaluate the combination of nematicides and soil surfactants as pre and post applications, and also to begin screening the varieties coming from Phil Simon's and Phil Roberts program. And then we will also be evaluating organic and conventional fungicides for alternaria leaf blight management. We'll continue doing the conventional and organic variety trials. And one of the additional trials will be the IR4 trial evaluating the use of Zidua pyroxosulfone in carrots. And with that, I would like to say thank you to Dr. Isolde Francis for providing us with the inoculum for our cavity spot nursery, my team, Jed Dubose, Jennifer, and Crystal Hernandez, and also funding from Bolthouse Farms, Fresh Carrot Advisory Board, and Grimby Farms, and my industry partners. And with that, I would like to say thank you. And if you have any questions, please put those in the Q&A. It limits the amount of soil fuming and say grower is allowed. No limits on the amounts allowed within a township, expanded buffer zones, or none of the above. And the next question is, which of these herbicides is a standard for the carrot industry?
Just brief, and then while that's going on, there's one question in the Q&A, and it's asking, in the future, can the private industry be included in the organic trial for the January evaluation?
Yes, for sure. And I will send out a notice when we request for seed. And in case the private industry wants to be part of the trial, that will be more than welcome. Yes. So the correct answer for the question first, the new regulations by DPR included all of the following except one.
Was it limits the amount of soil fumigants over?
And also if we can go over the second one.
Which of these herbicides is a standard for the carrot industry? And the options were Lorax, Dual Magnum, Glyphosate, or Outlook. And the answer was Lorax.
All right, thanks, Maria. I think we can move on to our next presentation. It is by Dr. Matthew Parade from University of Florida, and he will be presenting on the evaluation of fungicide performance delivered by solid set overhead sprinkler irrigation system on Alternaria leaf blight and assessments on cotton rot.
Thank you very much for the opportunity to give a presentation today. I just want to acknowledge the full team in Florida who've done and led the work, but specifically Sean Wang and Susanna De Silva, who are PhD students in my program. Also my collaborator, Bob Hawkmuth, who spent a lot of time working to configuring the system for us. Just to give a very big umbrella view, I'm going to talk about two diseases, Alternaria leaf blight and cottony rot. In case of Alternaria leaf blight, one thing many of you may know would be that it's a warm, wet weather pathogen. It prefers warmer conditions for occurrence of the disease and its spread. And when the right wet conditions follow, that is really ideal for its spread. The disease symptoms typically start as water-soaking lesions on the foliage. And also you could see it developing into small lesions with black or brown spots with yellow margins. And eventually that can lead to complete necrosis, as you're seeing in these pictures right here. And the disease can move quite rapidly within the system. When you think about Alternaria leaf blight, one of the key aspects pertaining to that is that if the leaf foliage gets damaged severely, especially at the harvest window, you're looking at decreased yields and decreased harvesting abilities because of the carrot tops not being available for the harvesters to easily pick it up. The two major pathogens that we know in Florida and in other places associated with the disease are Alternaria dowsii and Alternaria alternata. And their virulence can vary, but dowsii is the predominant pathogen defined in globally, but we are seeing more and more alternata being involved, probably less aggressive in that system, but still very important. Use of clean certified carrot seed, tolerant to resistant varieties when available, fungicide use in appropriate control and rotation to minimize resistance issues, and long-term crop rotation could be good tools to be used for management. The basic rationale of this study is that typically when plant pathologists and extension are looking at efficacy studies, we're typically looking at backpack sprayer system pressured by CO2 and looking at how fungicides are effective, and producers typically take that data and use that in the commercial system. But at the production level, if you were to think about carrot production in California compared to Florida, for example, in California, almost all of the fungicides are delivered through solid set overhead sprinkler system compared to, tractor mounted spray system, or in rare cases, some through the center pivot system in Florida, for example. So the key point is that fungicide performance data derived from a backpack sprayer system-based study, could that be similar to what you should expect in commercial systems? And we decided we want to actually ask the question pertaining to the two common delivery systems. One is the solid set and one is the tractor application. And how would the performances vary in this situation? We had plots with a 40 by 24. We had 4 beds per plot in one system, and we had 16 treatment plots in the study for alternaria that we did with 3 replicates per treatment. We had different checks, controls, everything with 2 application methods. And the way we delivered the solid set irrigation system, fungicide system, and the entire irrigation run through that same system, we had 4 rises, and this covered the center rises covered 360 degrees, and the end rises covered 180 degrees, essentially covering the full plot in the experiment. If you were to run the full irrigation system through the overhead solid set system, this is how it would look. It essentially covers all the beds. We did assessment to see what are minimizing factors where this might not be evenly spread, and we've been able to address all of that early itself before actually we started the fungicide spray program itself. But the system, while it took time, we were able to establish that. This is a picture of the entire field layout from a drone map image. And we used the variety Maverick for this purpose. We tested the products at weekly intervals. We did the disease assessment using Horsfeld-Brad scale. We had 3 separate individuals taking independent data and verifying, cross-checking the data. And what we did was, even though there were 4 beds within one plot and then 3 replicates, we took individual data within, and then we subdivided that also into further 3. And so essentially we had, a significant number of data sets coming in even within a plot. And the reason to do that was to ensure that earlier on we capture some differences that may show up. And we just wanted to ensure that structure. With the treatment procedure, we used treatments in case of solid set system. We pushed everything through using CO2 in the system and injected at 50 psi. And each of the treatment block irrigates at 40 to 45 psi. We did a system with a shutoff valve and a non-return valve to ensure that everything goes in and it's restricted within that treatment unit. And we would typically prime the unit until it flows well, and then we would push the whole system to make sure that it is a consistent run. And we would flush the system, and eventually, in case of tractor application, which is our comparative application, we sprayed at between 15 to 25 psi. These are the 16 treatments. Essentially, it's 6 treatments replicated in tractor application and 6 treatments replicated in irrigation or solid set application with water control for both and an untreated control for both scenarios, as you can see here. The application dates are shown here, and that's the application window. So we essentially had 6 application dates. Even though we planned for 8, we delivered 6, and based upon the growth of carrots and the disease incidence, we were mapping this as it went through. This is a graph which shows the area under disease progress curve. So what you're seeing in the y-axis is the abbreviation for that. This is a cumulative data of disease severity over a period of time. The orange bars that you're seeing, that's the data of fungicide effectiveness of fungicide in reducing the disease severity in tractor applied system. And on the blue bars are the ones with solid set irrigation system. If you were to look at your control, which is water and untreated in both cases, there were some differences, of course, in both systems. but it was pretty high irrespective. And we should note that the best products in this whole metrics was Merivorn and Fontelis in case of the tractor application system, and also Miravis Prime, followed by Nordox. But that was different in case of the solid set irrigation system where Nordox did not perform as well. However, it still reduced the disease in compared to the water control. And the best two products that did pretty well in the solid system was Miravis Prime and Merivorn. And Fontelis, while it did decently well, it did on comparative level with the tractor application, it did not perform as well. So this is a very interesting data set where we were able to show that the same fungicides could have differential responses in disease management when delivered through two unique type of application system. An important aspect to look at the end of the trial, for example, these are the plots that were treated with Merivorn through tractor application system. This is a comparative plot of untreated plots. And this is just to show the big field drone map, which showing that plot with the Merivorn and the untreated plot. And you can see, if you were to look at it, there are treatments that are doing well, and there are treatments essentially that have have very high disease severity. And we were looking at this mapping. And just to make it easy, there's a lot of data for all of you to take a quick look at this. One way to look at the results very carefully is to compare the actual disease severity itself, the percentage disease severity, and compare it in the sprinkler and tractor, and visually look at how the disease progressed over the period of time. So if you can look at all of these treatments, one of the most important thing that you would see here is that Merivorn, for example, was extremely consistent throughout and in its ability to manage the disease in both overhead solid set sprinkler as well as in tractor. In case of Miravis Prime, it also did pretty well with a little change at the 130-day window, 135-day window roughly, but it did pretty good. Now, if you were to compare Fontelis, though, that difference was not that there were more differences in there, as you saw in the prior data, where the sprinkler application had more disease severity. So the tractor performed better in case of Fontelis. Now, the big stark difference happened in case of Nordox, which is copper product, and where you're looking at where the Nordox delivered through the sprinkler system, of course, had much higher disease severity towards the end of the trial compared to a tractor application system. In case of Oso, which is a polyoxin-based material, this was a little surprise data because our multiple past trial data have shown that it worked really well. But we also think that based upon information also shared, some input shared by Grimway Farms during their visit, the rates that we used were not the highest available rate. And in fact, that may have contributed to comparatively medium level performance compared to past years. But irrespective, both the treatments look pretty similar. Both the application methods were pretty similar. In case of Stargus, again, there were differences. Now, if you were to compare Stargus, which is a biological control material, this flips actually compared to Nordox. In case of Nordox, the sprinkler system had higher disease severity, while in case of Stargus, it was the other way around, where the tractor application method. So delivery systems and the products, there could be significant variations in performances. And unless we really study how this all applies, it can be very quite confusing if you take a data from a backpack trial and trying to translate it to a commercial grower. So the important piece of information here is that depending upon whether tractor or a solid set irrigation system, depending upon the fungicides, there could be differences, and it really depends on the fungicide and the application method on its effectiveness. I want to shift gears to cottony rot. I mentioned before that in case of alternaria leaf blight, we are essentially looking at a warm wet weather pathogen. In case of cottony rot, on the other hand, which is caused by Sclerotinia sclerotiarum, it's a cool wet weather pathogen. And you can see yellowing of the plants. You can see yellowing of the plants during early stages of the infection, followed by plant death. The leaves completely collapse. And also you will find the black sclerotia on the ground, which forms the resting structures for future infections. And this is an important aspect in consideration to think about. The time of infection is cool, wet weather compared to Alternaria, which is warm, wet weather conditions. To begin this study, though, we had to establish in Florida, typically we don't see a lot of Sclerotinia-related issues, but we wanted to establish an approach towards using the inoculum. To begin with, in a literature review, we felt like maybe we want to look at Eulocladium-based isolates, which is Botrytstop was one product which contained this organism. And we wanted to see how that could, in a Petri plate, work against Sclerotinia sclerotiarum. So what my student, Sean Wang, did was to essentially plate Sclerotinia sclerotiarum, the cottony rot pathogen, in the middle of the plate. And it will grow, the mycelia will grow. And also there will be a, this Botrytstop at the center. And this zone indicates that it has potential to be competitive. It has potential to minimize the fungal infection, at least in the perspective of a plate assay. Our next step was to see how would it affect in a controlled detached leaf assay. So what you're seeing here in this plate is Botrytstop with Sclerotinia. So we have the pathogen and the product here. Number 2 is only the pathogen. And number 3 is only the Botrytstop. There's no pathogen involved. And as you can see, there's no symptoms exhibited here. And in case of number A, which is Botrytstop plus Sclerotinia, it appears, it's not very high difference, but it appears there might be some utility in compared to the pathogen by itself. And we were able to replicate this study a few times. When we look at the disease severity in this metrics, for Botrytstop plus Sclerotinia, when both were present in comparison to Sclerotinia by itself, you can see the trend of how the disease severity on that small controlled experiment worked. Sean actually went further to do a few greenhouse experiments where the trends seem similar. And we are currently doing field studies to validate this further. In this process, we also learned that we have to work on the inoculum a little bit. So we modified the existing oats inoculation procedures, which are published in different places and used by my colleagues at different places. We actually modified that to actually powder the oats which carried the inoculum and use that powdered oats for infection. This is just to show an example of how we were able to get the disease infect the plants by dropping powdered inoculum at the base of carrots within 0 to 13 days. And now this system is what we are using in the field trial, and hopefully in the next 2 months, we should have sufficient data that shows how this works. In conclusion, I would like to say for Alternaria leaf blight, Merivon, Miravis prime, and Fontalis are the most effective fungicides irrespective of the application method, whether it's a solid set or a tractor application. However, Fontalis and Nordox performed significantly better with tractor application compared to solid set. On the other hand, Stargus performed significantly better in solid set compared to tractor. So it really depends, which system could work. And it needs to be really tailored and tested out in multiple places, optimized application methods and rates, and potentially different carrot varieties to see how this might all pan out in a broader structure. In case of cottony rot, we have standardized a new powdered oats method for consistent production of the inoculum and infecting the plants. We have moved that from lab testing, greenhouse testing to now field testing. Based upon preliminary data from in vitro, some lab studies and greenhouse studies, we think Botrytstop might have a potential. Again, under field conditions, it could be a whole different scenario, but we are looking into that right now. And again, if we see that there's opportunity, optimizing the application and other methods will be very important. I would like to wrap up by acknowledging all the team members at University of Florida, University of Florida NFREC Live Oak, Ben, Mike, Dean, also the agronomist, Dr. Sudeep Sidhu, Darren Raj, who did all the drone flights for us. Also, I want to acknowledge Dr. Russ Hamblin, Jason Chandler, and Logan Petrie from Grimway Farms, who Jason and Logan took time to visit the plot, make an assessment, and provide inputs. California Fresh Carrot Advisory Board, and University of California Cooperative Extension, JAS, and others. With that, I'll wrap up and I'll take any questions if there's time. Thank you.
Dr. Parrott, there's one question in Q&A, if you would like to answer that.
So the question is, how many plants per plot did you use for disease assessment? We use entire plot for the disease assessment. So what we do is that in each replicate of the treatment, we divided that into 12 segments, and essentially each of those, and we use the risers as the guiding principle on where we start and where we end. But essentially we capture the whole plot, every single section of the plot. We are not missing any plant in the process. I see a question from Russ. Do you think that if fungicides is systemic or has locally systemic activity, it doesn't matter how you apply them? Do you think application method is more important for contact materials? I fully agree with you. Based upon this data, we are repeating this trial right now one more time. Based upon this data, it seems to me that the materials which are really systemic and have the greatest potential to work irrespective of the application method. On the other hand, contact materials, like for example, Nordox or for example, Stargus potentially, even though we may have to learn more about how it works, in those cases, application method certainly seems to be very important.
Question 1 asks, if a chemical is labeled for tractor applications and for irrigation applications, would tractor be expected to be better than solid set irrigation for application of fungicides from carrots for Alternaria leaf blight management? And then question 2 asks, you have a field with confirmed quaternary rot and Alternaria leaf blight infections. For the past week, field conditions have been wet with average daily temperatures of 85 Fahrenheit and rising. Which disease do you think will be most likely to cause damage? Matthew, did you want to go over the answers or would you like me?
I can do that. So that for the first question, the answer is it depends. It goes back to application method. It goes back to potentially the systemic nature of the materials. So it can really depend and vary depending upon a variety of factors. So the answer is it depends for that first question. For the second question, the answer is Alternaria leaf blight. Essentially, you can think about a products that would potentially have effect on cottony rot to be applied in a system when it is cool and wet. When it transitions to temperatures of 85 Fahrenheit or higher, you're really looking at that infection slowing down, and it could still cause damage because at the level it already happened, but really Alternaria picks up big time at that point of time. So I think majority of the people got the answers right.
Thank you, Dr. Parrott. And now it's time for our first guest speaker, Dr. Linda J. Harris from University of California, Davis. And she will be presenting and providing us updates to the Produce Safety Rule proposed revisions to Subpart E, Agricultural Water. Dr. Harris?
You will be have access to a recording, and you will also have access to the slides. So the information here, if you need to go back and review something, will be available to you. I gave a presentation at this webinar 2 years ago, right after the FDA had proposed their revised rule for Subpart E or for agricultural water. So that was published in December of 2021. At the time that I spoke in March of 2022, the comment period was still open, and the comment period closed April 5th, 2022. You can go online and have a look at all of the comments that were posted. There was over 130 comments that were posted. The last time I gave this presentation, the last slide I had was a question, when will the final rule be published? And basically, it is normally, if there's no substantive comments received, less than 60 days or less from the close of the comment period. If there's substantive comments received, then of course it's a little bit less clear. I used the full produce safety rule as an example. Between the proposed and the final rule was about 3 years. And of course, that was a much, much larger rule. So I had guessed that it would be more than 60 days, but less than 3 years. And we are currently at 1 year and 11 months. The rumor is that the final rule will be published in March. I checked this morning, and it was not published as of today. So soon, but how soon, I cannot say. So I'm just going to briefly go through what the proposed rule covered and what the differences were between the original and the proposed rule. But to get everybody on the same page, agricultural water has always been in the original rule and in the proposed rule that agricultural water must be safe and of adequate sanitary quality for its intended use. And so agricultural water is that water that is intended to or likely to contact both covered produce or produce contact surfaces. Produce contact surfaces are mostly harvest and post-harvest handling. So really the part that was changed in the proposed rule is the pre-harvest water, not the post-harvest water. And this would cover irrigation water. It would cover crop protection, water used for crop protection, spray application. So all of those types of water in the various ways in which they're used that are likely to come into contact. Now, carrots are kind of unique. I mean, there are other root crops, but the fact that they have a harvestable portion above, which is the leaf, that's part of what is harvested. And then, of course, the carrot itself is below ground. So in some crops, drip irrigation might not be agricultural water. If you think of tree fruit, then drip irrigation would not reasonably come into contact with the product unless the drip line was broken. But in the case of carrots, you're putting water into the soil. It's intended to move over to the carrot, and therefore drip, overhead, tractor applied water application would all qualify when you're talking about a carrot. So carrots are covered produce, and drip irrigation, as an example, is likely to contact. As you notice, I forgot to mention earlier, these slides were developed by the Produce Safety Alliance in their supplemental material after the proposed rule was proposed. And so I am mostly using their slides for this presentation. So the current rule, as most of you should know, depended, on a definition of adequate sanitary quality that was based on prescribed testing frequency and then calculation of a geometric mean and a standard statistical variance and of generic E. coli. And there was both pre-harvest and post-harvest testing requirements. The proposed rule made the biggest change in that although testing is still a potential part of what you do to make an assessment, they really changed the approach to a more holistic approach, which would be to do an agricultural water assessment, which is a review of the whole water system. And that each grower then would set standards and would try to understand what those standards provide or how those standards provide water that is safe and of adequate sanitary quality. And when an inspector would be coming to on site, the conversation should be not only to explain what the water agricultural water assessment was, what was found, and how things are being controlled on the farm to an adequate sanitary quality. Now, I'm not going to go in detail through all of this, just to say, again, you'll have access to these slides. But in an agricultural water assessment, these are the things, the part of that holistic approach. You're looking at the whole water system, which is source, distribution, degree of protection of that water source, the practices that you're using, the time interval between the application and harvest, crop characteristics, if there are data to support less susceptibility or more. And then environmental conditions, frequency of rain, air temperatures, UV exposure. And then, of course, other factors could include having a data set of results from testing of water. And I think, different growers are going to be in different places depending on the approach they've taken in the last couple of years in dealing with agricultural water while they've been waiting for the final rule to be published. So in this assessment, which in a couple of slides, you're going to see the AWA, the agricultural water assessment. If it is determined in that assessment that the water is not safe or is not of adequate sanitary quality, then there has to be an immediate discontinuation of the use and then corrective action measures taken before harvest or before use in pre-harvest. And then, there is specific language around known or reasonably foreseeable hazards, which include animal activity, BSAAO's biological soil amendments of animal origin, or human waste. And then a category on one or more known or reasonably foreseeable hazards not related to the ones that I just mentioned. And then, of course, at the other end of the scale, no known or reasonably foreseeable hazards. And then, you would inspect the water system regularly. And you can define what regularly is, but at least once a year. So all of these have, and if this is the case, then you must do this. So again, this comes in as a overall assessment of your water system. So some of the examples of corrective measures might be reinspection of the entire system and making changes that are necessary to get things back into control, treating the water in accordance with the standards in the produce safety rule, the existing produce safety rule. Some mitigation steps might be making repairs, increasing the interval between application and harvest, because there is an expectation for microbial die-off the longer the time between application and harvest. Taking credit for die-off during storage, which might be applicable to some crops. Commercial washing might be applicable to some crops. You need data to support that. And in the carrot industry, certainly post-harvest washing occurs. So this might be something that could be used with the appropriate data. Changing water application methods might not be as applicable to carrots. Treating the water and taking alternative mitigation measures that are supported by data that you have either generated yourself or that come from other scientific studies. In March of 2022, so 2 years ago, almost to the day, FTA posted its Ag Water Assessment Builder. And so this is something that you can go to and use. It'll step by step help you through the process of building your agricultural water assessment. I assume that if there are changes in the final rule from the proposed rule, that this would then be updated. And I assume, again, that once the final rule is published, there will be a lot of training that will go on in using this and helping to growers to build their agricultural water assessment. As I mentioned, there are corrective measures, mitigation measures, which I think I already mentioned most of these. There's differences between the final rule and the proposed rule. They give you actually a little bit more options in the proposed rule for dealing with corrective actions if they're necessary for your water. So just a brief comment on water treatment. It's always been an option, and it's still the case within the proposed rule. You can use the E. coli populations measurement testing as a way to drive some of your decisions in what you're going to do, and to ensure that the treatment is actually working as intended. There's a lot of different water treatment chemicals, a lot of work going on in this area, especially with the leafy greens industry. They're, I think, further ahead than most produce crops as far as looking at effective treatments for water that are being applied to crop. So this slide is from Channarok at the University of Arizona, but there are a lot of data, I think, coming out that would be applicable if this is something a carrot grower wanted to do on their farm. The information is going to be out there for treating water. Okay, so the one caution here is that, if you're using a crop input and using the water for application of that crop input, you need to make sure that whatever you're doing to treat the water is not going to make the crop input inactive or change its effectiveness. So I mentioned that there's a need for a reassessment of agricultural water. This needs to be conducted at least every year. And anytime there's a significant change in the water system you're using, the practices that you're using, a change in the crop that you're growing, a change in the overall environmental conditions, something has happened that wasn't there when you originally did the assessment, and it might change the risks especially to a surface water source. And in this reassessment, you need to, look at those changes and those new hazards. And with anything that's worth doing, there is a requirement for record keeping in the case of this agricultural water assessment and the reassessment. So during the initial assessment, you need to have a description of the factors that were evaluated, written determination of whether corrective actions are needed, and then what you did to bring things in control. And if you've been testing, you can use this as part of the assessment to give some kind of information on the consistency of the quality of the water that you're using. Supervisors must review the written pre-harvest agricultural water assessment and the determinations. And of course, the only way you would know that they had reviewed it is if they sign off on that review. Now, there are some exemptions. I don't know how many of these exemptions would apply to your specific situation, but if you are using an untreated groundwater and you have test results for generic E. coli, so an absence of generic E. coli from those test results, then you might be able to use that data to be exempt from the pre-harvest egg water assessment. If you're using public water, so that's sometimes the case for some growers, and so that would be a reason for an exemption. Keeping records from the public water system of their testing would be something that you would need to document. And then again, if you're treating the water using the existing standards. So once the rule is finalized, when do you have to put this all into practice? So the larger businesses, it's 9 months after the effective date. And then small and very small businesses have either 1 or 2 additional years to comply with the final rule. These fact sheets were published 2 years ago, so they are still available and would provide some background information that FDA had written at the time. I would imagine that these would be promptly updated once the final rule is finalized. And then I want to also point you to an industry publication. This is from the International Fresh Produce Association. I put the URL down at the bottom, but honestly, if you Google International Fresh Produce Association and then the title of the document, you will get there probably quicker than trying to capture this link. And so that is something that I think would be very useful to anybody trying to implement an agricultural water assessment. So with that, yes, Jaspreet. Okay, thank you.
Thank you, Linda. And if there are any questions, I'll pass it along.
Perfect, thank you. Bye-bye.
Thank you so much. So if you have any questions, please type those in the Q&A and I will collect those and pass it along to Dr. Harris, and we'll get a response from her. Next speaker is Dr. Meredith McDonald from the University of Gulf, Canada, and she will be presenting on screening carrot lines for resistance to cavity spot and other traits.
Yes, thanks, Jaspreet. I just want to begin by pointing out that this work is all done in collaboration with Phil Simon, and graduate student Umberin Ilyas has contributed to some of the work I'll be presenting also. Some of you may know that this work is all done in Ontario, Canada, so we're about 30-minute drive north of Toronto. We've got very high organic matter soils. You can see the black soils here. And cavity spot occurs every year, sometimes more severe than others. And the carrots are usually seeded in late May or early June. So not too different from the trials that Jaspreet was talking about earlier. And of course, our objectives are to work with the USDA breeding program to select for resistance to cavity spot with the long-term objective of contributing to the breeding program for carrots in California. We always include at least one highly susceptible carrot to make sure that there is cavity spot in that site. Atomic red, a red carrot, has been the susceptible check for many years. And a number of the dark purple carrots are quite or totally resistant, and we've been using deep purple for a while. We include some of the cut and peeled carrots that you might be familiar with, and some of the cello pack carrots as well. And there's more than one Pythium species that causes this disease, just to keep things complicated. In California, Pythium sulcatum and Pythium violae have been found in trials over the years. We always say that sulcatum is the main species in Canada, and it is also in Washington State. And recently, the grad student did an extensive survey with well over 100 isolates collected from our plots as well as grower fields. And in addition to sort of the traditional culturing, these were also sequenced to confirm the identity. And sulcatum was definitely the most prominent one. But we were kind of happy in a way to see that violae was there as well. That makes it very consistent with California. And Pythium violae was found in all of the fields that were tested. And then there's sort of this group of usual suspects that have been reported over the years of being isolated from cavity spots. So our methods have been consistent all along, so we can compare from year to year. At least 60 lines, all direct seeded on raised beds in our high organic matter soils, 4 reps for each line, and 6 meter or 20-foot rows. And of course, we don't apply any fungicides that could affect cavity spot, but the rest of the production is consistent with what the commercial carrot producers would do. So we already heard a good introduction from Dr. Pratt about alternaria. We sometimes see Cercospora on our carrots as well. We assess these together, and we also look at forking. The forking can be caused by Pythium, as Pythium root dieback, but we're also aware that there are lots of other things that can cause forked carrots. So we harvest in mid to late October, assess 50 carrots for cavity spot, another 50 for forking, and we put them into classes based on the size of the largest lesion on each carrot, and then calculate the disease severity index. Our leaf blight is assessed on a 0 to 5 scale, where 0 is healthy and 5 is basically dead because of the disease. The important point is that a rating of 2 or lower is considered a good rating in terms of low carrot leaf blights. In 2023, the stand was reasonable. We had excellent stand in 2022, not quite as good, but all of the lines certainly had enough carrots for us to assess. And cavity spot was higher this year than in 2022, similar to 2021. So the highest severity was around 50%. And we did have to irrigate in August. We know that higher soil moisture contributes to cavity spot, and it was getting pretty dry, so we put water on the trial. Leaf blight was moderate, and forking was actually lower than in many years. So here's our results from 2023. Of course, we assessed the whole 60 lines. I'm just showing the low cavity spot lines, and then also some of these ones with that are very susceptible to cavity spot, including atomic red, but there's also a number of orange carrots that are also very susceptible. But we do have to have those susceptible lines, so we've got a good comparison with the others. Deep purple, 0 cavity spot this year. But the good news is we had many orange carrots that had low cavity spot, less than 5% severity in this case, and many of them right around 5%, definitely under 10, which is very promising. Some of these, the ones that start with NBH, are also resistant to nematodes, and you'll be hearing about those later on this morning. And then these F77 lines have been consistently showing low cavity spot over the past number of years. And there are a few commercial cultivars that are also relatively low, but unfortunately, some of them are also in this grouping of very susceptible to cavity spot. So just to compare to 2022, again, overall lower cavity spot, but fortunately, very much the same trend with this group of crosses with the nematode resistance and with a couple of these parental lines that are showing good levels of resistance. And again, just 2021, all the same trend, which is really good. We're happy to see this consistency in the results. So it's always interesting to see what these carrots actually look like. And this is how we do our assessments. This is the batch of 0 cavity spot carrots, and then we divide them into those other 5 classes. This one had the lowest cavity spot in the 2023 trials. So very few had any cavity spot at all. And this was another one with very low cavity spot. So you can have a look at how that carrot, what that carrot looks like. And again, hardly any in with any cavity spot. Of course, the deep purple, it is a very dark purple carrot, no cavity spot. And just as a comparison, this is the atomic red. It looks more orange than red here, but it is quite red. And I hope you can see that these ones in the high category really do have some large cavity spots. Hardly any of the carrots had no cavities at all. And then this is, again, one of the orange carrots that's highly susceptible. Hardly anything in the no cavity spot category. So just to show our leaf blight ratings and forking, again, deep purple, no cavity spot. Here we have the nematode resistant crossed with some of the other lines. And if we go down here, we did have one of the parents with very low leaf blight. And again, to compare to 2022, we also had a cross with one of the more resistant cavity spot parents crossed with the nematode resistant, and again, very low leaf blight in this line. So to wrap this up, we did have higher disease pressure this past year. We're seeing several of the orange lines with low cavity spot, which is always good. And it's even better to see that some of them have nematode resistance as well. We did have one of the lines that also had low leaf blight. Again, always a positive thing. There are lots of cavity spot susceptible lines out there, and it is important to have these in the trials as well. And of course, this all contributes to Phil Simon's breeding program. And I know we're going to hear from Phil coming up next. So our reports are published online every year at the website. The 2023 data will be up in a few weeks. just want to acknowledge the team that we have working on the research station last summer. And of course, to thank the funding provided by the California Fresh Carrot Advisory Board. And we also get some funding from Canadian groups, especially to support the work that the graduate student is doing. So I hope there might be time for a couple of questions.
Yes, we have a question in Q&A, Mary.
In the Q&A.
Yes.
Oh, the publication slide again. Sure. Yeah, are the deep purple carrots less susceptible or you can't see damage? That is a good question. We get asked it fairly frequently. We really are convinced that they are less susceptible, partly because Phil Simon in the past has had some purple or purplish carrot lines that were susceptible to cavity spot. So I know it's a question that comes up quite a bit, but we are convinced that they are resistant. And the publication slide, I'll see if I can get back to that.
With that, I'm going to launch the poll. There are 2 questions. The first question is, which color of carrots is generally most resistant to cavity spot? And then the second question is, true or false, carrots that are resistant to cavity spot are also resistant to lithium group dieback. To end poll and share the results. So it looks like for the first question, most people got purple, and that is the correct answer. And for the second one, most people put false. You guys got it right. That one is false.
Thanks, Dr. McDonald and Maria. So we'll go on to our next presentation by Dr. Phil Simons. And he will be going to present on the carrot breeding to develop and introduce improved cultivars for California production. Phil? Okay.
Thanks for your introduction, Jas. Let's see. Well, I appreciate the opportunity to speak to the group today, as well as the continuing support from the California Fresh Carrot Advisory Board. The carrot breeding program has a number of different directives in terms of the types of carrots that we're developing, and you've heard about some of them from Jas and Mary Ruth, and you'll hear some more from other speakers. The program depends on these collaborators to help us identify new sources of I'll particularly be talking about disease resistance today. And these are the primary collaborators on this project, as well as individuals from the industry. Let's see how I can advance this. There we go. So the scope of the breeding program is fairly broad in that we focus on what we have come to understand are the major challenges to carrot production from the grower industry. And we work closely with seed companies since they're the conduit through which the new carrot varieties are developed. You've heard about some of the field trials that have collaborated with the carrot breeding program, and the list of the collaborative field trials is here with a major trial in Southern California, Desert Research and Extension Center in El Centro near the Mexican border, as well as trialing in the Central Valley. Those trials are with Dr. Sidhu, where we evaluate general breeding materials. We also have trials in the Coachella Valley with Phil Roberts, as well as some greenhouse screening to evaluate nematode resistance. You heard from Mary Ruth McDonald, who's collaborating with us in identifying new sources of cavity spot resistance, and also from Jas Sidhu on her evaluations of cavity spot. The alternary leaf blight resistance screening are primarily doing in Hancock, Wisconsin, where it's a natural and infective agent, along collaboratively with Irwin Goldman. But as you heard from Mary Ruth McDonald and from Jas Sidhu, we also have some evaluations for these diseases by these collaborators. From these field trials, carrots are typically sent back to Madison, Wisconsin, where my project is located, where we will take those roots that have been evaluated and evaluate them also for things like flavor and nutritional quality, and then use those carrots for seed production. This occurs both with the trials from Jas Sidhu as well as Dr. Roberts and Dr. McDonald's programs. So it's very important to have these collaborations for us to make progress in the breeding program. I won't say much about it other than this slide, that we also, beyond the diseases and pests that are the major focus of this talk, we also evaluate things like fresh carrot flavor. This is a fresh carrot breeding program, and we want to be sure that the carrots we develop are edible. So we look at flavor. We look at color as a part of the appearance as well as the nutritional value, and uniformity. And these are things we do in the lab and in the greenhouse and fields there in Wisconsin. Another part of the program I won't say much about, but it's critical to improving the crop, is developing new DNA markers to track the genes that control the traits like the nematode resistance, for instance, that we'll be talking about in the program. A very brief slide on where we are getting genes for the traits I'll be talking about: nematode resistance, cavity spot resistance, and alternary resistance primarily. We look at this slide, we get most of the genes from carrots in the middle of this slide, where carrots on the left are wild carrots, which you can intercross with cultivated carrots, and they are important for sources of alternary resistance. But for nematode resistance, we're primarily getting our resistance from this group in the middle. None of the modern cultivars on the right of this slide have any significant nematode resistance, but luckily there's some very strong resistance in this middle group, which we would call heirloom varieties or foreign cultivars. For something like the cavity spot resistance, we do find some fairly good resistance in the modern cultivar types, but also from some of these heirloom and foreign cultivar types. So that gives you a little bit of idea where these genes are coming from. We intercross carrots with these resistance genes to the modern cultivar types, improve them for other traits, and develop new breeding lines for the seed industry. So starting with some of the general trials this last year with Dr. Sidhu, we always include some experimental hybrids in the trials, in the El Centro trials, and we do this to make sure that the carrots that we're developing are not only have the traits that we're interested in, but also have the correct appearance for the growers of the crop. And I'm pleased that we have a number of entries of experimental hybrids from the program that scored well in the general evaluation of carrot appearance, like the carrots you see on this picture, where they're nice and uniform and good colored and well shaped. So we have some good experimental hybrids in the program, and some of these have very good nematode and cavity spot and alternary resistance. That's the general trialing. We do this primarily in Southern California. California is by far the major carrot growing region of the US, but we also do some trialing here in Wisconsin because you get some additional information on alternary resistance there. In the nematode trials in the last couple of years, starting in 2022, we've had some evaluation of some new sources of nematode resistance. In the last year, in 23, we had 580 entries on a Meloidigani incognita trial, and Dr. Roberts will be talking more of this. We have advanced the evaluation of some new sources of nematode resistance for this Meloidigani incognita species, as well as confirming some of the earlier results. And we also had a field day this last year in this 2023 trial We were unable to have them in earlier years with COVID, but now we've resumed the field day where we invite growers, processors, and seed industry participants to see the trial in the nematode-infested field. Looking more in a little more detail at the nematode-resistant carrots, we historically have relied on Brasilia, which is a cultivar that's similar to the carrot you see on the left of this slide. It's rather short and thick in diameter. We found some good nematode resistance in this cultivar. I'll show you a slide about how about that in the next slide as well. But we also have been moving the traits from this heavier carrot into longer, thinner carrots for the cello carrot industry and the cut and peel industry. And again, this Brasilia cultivar has a number of very good resistance genes, and we are we've released some resistance to the seed companies, and they're using it for improving their carrots. And we're in the process of releasing some additional carrots, including 2306 listed here, which Mary Ruth McDonald mentioned in her slides with cavity spot resistance work. Cape Market's a cultivar from South Africa. It's a new source of resistance where he's evaluating the genes for Cape Market resistance as part of the nematode resistance breeding project and incorporating those genes into cut and peel inbreds in the experimental breeding program. I will mention that as the seed industry is using our sources of nematode resistance and also pursuing some of their own, we give them a chance to trial their carrots in our nematode-infested fields. And we've had 6 seed companies in the last year putting entries in that field, and several entries from each of the seed companies have very strong scores of 0 or 1 on the 9-point scale. Indicating that they've got some good disease resistance being developed, and these are headed for finished cultivars in the next few years, I've been told by them. I'm not going to go into any detail on this slide other than to point out that in addition to the resistances from Brasilia, we also have genes coming from an Australian cultivar, Western Red, a cultivar from Syria called Hams, a Chinese cultivar called Pingding, and a couple of European cultivars, Scarlet Fancy and Nance, as well as the Cape Market. So we are looking at combining genes from all of these sources of resistance to make a more nematode-proof carrot as we improve the crop for the California industry. And down on the bottom, you can see we've got some intercrosses with carrots that are good performers for the cut and peel industry, as well as carrots with good flavor and resistance to some of the other diseases besides the nematode resistance. So the original identification of resistance from Brasilia is demonstrated in the left slide, upper left slide here, where you see Brasilia grown in a nematode-infested field. And Phil Roberts is a nematologist, and he can tell you more of the details, but but you can see the variation in nematode resistance in this plot. Some are quite resistant, some are susceptible. We take the resistance ones and cross them with susceptible cultivars that we're breeding for California production to incorporate those genes. In the middle of this slide, we see some of the lines of carrots that we're using to bring in that resistance from different gene sources, and some experimental cello hybrids is 8523 and long cut and peel 8507 hybrids that are quite attractive and perform well for the California industry. Moving on to cavity spot resistance, Mary Ruth McDonald gave a good background, and as did Jacido, on the breeding for cavity spot resistance. And we're pleased to see that these lines F7737 and 7738 continue to perform well on several locations, both in California and Canada, but also we have some trialing in Washington where they're also performing well. We do the seed production, produce the seed, and then send it back to Mary Ruth and Jas and Phil Roberts, and they do the continuing evaluation for resistance. In this case, it's Mary Ruth and Jas for cavity spot resistance. As noted, 2306 is also resistant against nematodes as well as cavity spots, so that's a very promising inbred. And again, we're interested in putting together these different sources of gene resistance in the same carrots to make them more durable in their resistance in the long run. Moving on next to alternaria leaf blight. This is trialing we're primarily doing in Wisconsin, but certainly relying on results we get from Mary Ruth McDonald and Jas Sidhu, where there's also an incidence of alternaria leaf blight. And we use that information also for breeding in those genes for resistance into the new cultivars or new breeding lines that we're developing for cultivar production. And again, we're happy to see generally consistent disease ratings in Wisconsin and California. In other words, a carrot that many of the carrots that are more resistant to alternaria leaf blight in Wisconsin are also more alternaria leaf blight resistant in California. I should have included it here, but also in Canada. And we're intercrossing these sources of resistance to develop more alternaria leaf blight resistant carrots for the carrot industry. Just a brief picture of the seed production. Again, when we get the roots from these trials, we put them in a refrigerator to stimulate floral development, flower development. We do some of that production in the greenhouse, some of it in the field, and produce seed, and again send that seed on to the collaborators. Coming up, we aim to continue these collaborative efforts and develop more and release more breeding lines for the traits that I've talked about today. Again, we breed for other traits as well, like flavor and shape and nutritional quality. We're interested in tracking these genes to help facilitate and speed up the breeding process with genetic markers, DNA markers, because this makes it for a more efficient breeding program. And in closing, I'd like to express my appreciation to the California Fresh Carrot Advisory Board for their ongoing support, and also to you for joining us today. So thank you very much.
Thank you, Phil. And we have 2 questions in the Q&A. Phil, if you would like to answer.
Yeah, if you could read them to me. I was looking for their chat box and I'm not seeing them. So
yes. So the first question is from Russ. What level of nematode resistance would be required for commercial carrot seed companies to be interested in incorporating some of your resistance varieties or genes into their commercial offerings?
That's a great question. It's hard to put an exact answer on it. Like most things, the more the better. We do see that with crosses with some carrots like the 2306 line, NBH 2306, that has been mentioned several times, probably gives enough resistance to give you a very consistently nematode resistant carrot as a cultivar. So generally speaking, having one source of resistance may not be enough, but I would say that so we have had experimental entries from the seed companies in our trials in Coachella, California. We see some quite good resistance there. We don't know exactly what the carrots that the seed industry is giving us are. We leave it to them to spread that information to the growers and others. But we see very good resistance. I'm optimistic it's coming along fairly well. Russ, keep asking the growers, the seed companies, I should say, when they're coming out with their cultivars.
Thanks, Phil. And the next question is, does higher sugar content translate into higher likelihood of root disease problems?
That's a great question. We always are worried about is some of this disease and pest resistance going to bring in some poor eating quality or poor growth, or let's say a rough carrot surface. Luckily, we found very little problems in any of these disease resistance and pest resistances conferring any reduction in the quality or productivity of the carrot. So keep our fingers crossed. That's where these molecular markers, these DNA markers, are helpful. In some cases, the resistance genes are close to genes for flavor and color and productivity. But as we track them in the breeding program, we're able to develop carrots with both these improved disease and pest resistances, as well as good eating quality and productivity for the grower.
Thank you, Phil. And Maria, if you could please launch the poll.
Which of the following is true? And the correct answer was the first one. Symptoms of the cavity spot disease in carrots become more pronounced after long-term post-harvest storage.
Great. Thank you so much, Phil.
Thank you.
And we will move on to our next speaker, continuing on the same topic. Dr. Phil Roberts from University of California, Riverside, and he will be presenting on identification of gene sources for resistance to root knot nematodes attacking carrots in California.
Okay, good morning, everybody. Pleasure to join you and share a brief update on our nematode resistance work in carrots. I've been working obviously very closely with Phil Simon's breeding program in Wisconsin, and Phil talked quite a bit about nematode resistance, so I'll sort of fill in the gaps and give a bit more of a detail on how we're approaching this. I'd just like to give a mention to Bill Matthews, who's in my lab at Riverside, who does a lot of the legwork on field and greenhouse assessments for resistance. So I've shared this with you before. Some of you are new. Just a pictorial of nematode infection on carrot roots. The nematodes on the lower left here. The worm-like juveniles get into the young roots, and then they develop and cause galling tissue around their feeding sites, both on the feeder roots and on the taproot, which is the main economic damage to carrots that root knot nematodes cause. These little juveniles develop into large round females, and the females produce egg masses with several hundred eggs. So for the carrot, it's a question of numbers. Many nematodes on a root can cause a lot of problems. Phil introduced where we're getting nematode resistance from, and I've just, we have many sources identified now, which is really very positive, including some sort of next generation or new generation materials. I've just highlighted in red here some of the key ones, the Brasilia 1252, which are in the NB lines, our most advanced materials, sources of HOMS that we've selected and crossed with Brasilia. So those would be NBH lines with high incognita and javanica resistance. These are two of the predominant Meloidogyne or root knot species that attack carrots. We also have HOMS selection with very strong Meloidogyne haploresistance resistance. And I won't talk more today, but we did have a field trial up in Washington State this last year where we got some of the haploresistant materials into the field for the first time, and some of that looks very promising. So the key here is using single sources, but also in these crosses to look at combinations of resistance, and how this gets back to Russ's question just now, how effective that resistance is, the level of resistance, and also how likely it is to remain effective where nematodes are not getting around resistance or breaking it with virulence. So this year we had two, we went back to our Coachella Research Station farm, we call CVARS here, in Thermal, just east of Palm Springs. And we have again used the M. incognita site that's heavily infested and really has done fantastically well in identifying truly resistant lines, and also within those lines individuals that are where we have segregation, individuals that are resistant. So very powerful. And I'll just mention we have a new site at CVARS that is ready now for trialing with M. javanica infestation. And I'll touch on that again in a minute. So that's a new development. So for the 2023 trialing, this was planted end of March, and we harvested at the end of August. We were going to harvest an assay a bit earlier, but we had a terrible monsoonal washout at the station. Very fortunately, it didn't affect our carrot trials there, but it caused huge damage to other experimental plots. So weather's very unpredictable. Here's a view of the trial on May 12th, 41 days post-planting. We had pretty good stand. You can see some lines fail for whatever reason in terms of having poor germination, but overall, very good germination and stand establishment. Here's just a shot to remind me to mention this new javanica site where for the last couple of years we've been planting host tomato and lima beans. This is where it's just being brought up last year to create heavy uniform infestation and we will have some trialing there this year. So back to the incognita field, we did the harvest and the root assays at the end of August, and as Phil had mentioned, we did have a field day for the first time in a few years where we had some of our industry stakeholders and partners come in and have a look at what we had going. We typically grow on the left here an imperator susceptible check every 5th plot in our plot layout, and can then contrast. Here's a resistant line looking very clean. We can contrast the level of resistance across the plots. And as Phil mentioned, we've typically brought selections from previous trialing. For instance, going back a few years where we've trialed at the South Coast field station and the Kearney field station up in the Central Valley, bringing in resistant selections like this one on the right here compared to a susceptible Imperator 58 on the left, bringing those forward and retesting them. And some of that material was in the 2023 trial again. So in the submitted report to the California Carrot Board, there's a long table that we put in there documenting all the results from the field trialing. We had a large number of plots, nearly 800 experimental plots and plus several hundred industry submitted plots, and hundreds, probably thousands of roots selected to ship back in this shuttle breeding effort with Phil Simon, shipped back to Wisconsin. And of the advanced materials, we're seeing somewhere around 60 to 80% of the advanced lines with very good resistance. So this is scores of less than 2, many rated as 0 on a scale of 0 to 9, where 0 to 7 rates the root galling, and then scores 8 and 9 score if we have rot coming in from other pathogens, and we had very little rot this year. So our Imperator checks, a mean score is nearly 1 of 7. They're just really a hot infestation, and so here we're also looking at many early generation progenies and as Phil indicated, trying to select for good horticultural quality as well. So in the table one is a sort of a listing and description for those, particularly the seed companies interested in the materials. We had F3 and later generation materials selected from our 2021 field trialing. We had a lot of early generation F2 populations with resistant sources, Brasilia Homs or Homs, cross with elite susceptibles. We've included a breeding pool and other lines from some of our associated USDA project materials. This CIOA is an organic ag focused project, and in there some of the NB, so Brasilia source lines showing good resistance. Lot of new breeding populations with high resistance, advanced breeding populations selected out of the last few years. And then very importantly, these hybrids that are listed in the report with the Brasilia Homs or Brasilia resistance as donor parents have, and Phil touched on some of this, have been looked at in the field and look really strong, and also retested in the greenhouse. So just to finalize, we do a lot of greenhouse testing with controlled inoculations. Here's our setup in the greenhouse. So we squirt nematodes into inoculum into these pots and then assess the roots. And we focus primarily this year on this Cape market source of resistance, and 2 or 3 plant introductions that look extremely promising, with high resistance and also resistance to multiple root knot species, which is obviously very desirable. We've reselected out of 7 released resistant inbreds, so which will then be put back as stocks to ensure their resistance levels. And then we've looked at these 10 best parents, screened those again for hybrid releases, and Phil touched on a couple of those. So it's an iterative process, but I think we're making good progress, and it's up to our seed company partners to actually push out nematode resistant varieties for growers, but we're very close, I think. Our planning for this year at Coachella is scheduled for next week, both on Incognita and Javanica sites, and just like to acknowledge the Fresh Carrot Advisory Board support, our different USDA funded programs, and then our seed industry partners. So thank you very much.
Thank you, Phil. Maria, could you please launch the poll?
Yes, so there's 2 questions. Which of the following is true about field carrots with nematode resistance? And the second question is there one species or multiple species of root knot nematode that attack carrots? So the answer for 1 was the second one. There are currently no available commercial carrots with nematode resistance. For the second one, the answer is multiple species.
Yep. All right. Our next speaker is Dr. Steve Fenmer from UC Davis, and he will be presenting on steam disinfestation of weed seed banks in carrots.
All right. Good morning, everybody. Chance to speak to you again. I'm going to talk to you about some results that we have created over the past couple of years, and I'm also going to tell you about a new machine that we have built. I was over in Kern County about 2 weeks ago and got some information about the needs of the industry, and we are going to respond to that. We are designing a bed shaper that will conform to the needs of the carrot industry. So that's what we're working on. So I'm going to go over a couple of sets of results that we did in carrots a couple of years ago. I didn't apply for funding from the Carrot Board in 2023 because we weren't ready, and I have applied again because we are, I believe, now ready. So what we started out to do is to evaluate soil disinfestation with steam for control of diseases and weeds. After the meetings a couple of weeks ago, it sounds like there's much more need for weed control, and that's something that I'm pretty confident that we've been able to provide. And we're looking at the ideal conformation. But based on the information I've gotten recently, it sounds like we need to treat the entire bed top, focusing on organic carrot production. So soil disinfestation is sort of a blanket term that you can use to talk about steam and solarization and fumigants. And so it basically is a kill step, is a treatment that that reduces the soil pest infestations to something that you can live with and hopefully remain profitable. And so there are three theoretical patterns to apply steam. The top one, you can treat the entire field to the desired depth. And that photo of the John Deere and the big train behind it is taken from a strawberry nursery in far northern California. What I'm talking about today quite a bit is band steaming, where you strategically target the planting area. This is, and I'll explain what the process in the next couple of slides. And then the ultimate in precision is spot application, and that photo at the bottom there on the right is showing that process in a experimental method in strawberries. So how soil steaming works is you inject steam into the soil to raise the temperature to 158 for 20 minutes. The steam injection does not need to last for 20 minutes. The heat needs to last. And what we have found, I've been working on steam since 2007, is when we get soil warm up to the target temperature, it cools slowly enough that we get that desired interval or dwell time. And so what's happening in the soil is that the steam transfers the heat. So when you condense water, you're imparting a tremendous amount of energy. That's, in this case, from either diesel fuel or propane. You transfer the heat to the water vapor, and then the water vapor is simply used as a fluid mechanism, a vapor in this case, to inject heat into the soil. And so when the water vapor comes in contact with the cold soil particle, the heat, the energy from the steam transfers to the soil particles and releasing the energy as the vapor condenses into water again, returns to water. And so that heat is what kills the pathogens in the weed seed. And so when you're killing weed seed or nutstitch tubers, in the case of seeds, you're killing seed embryos, and they do not need to be germinating. You can kill them in their ungerminated state, dormant or not dormant. And so this is what we're doing. This is maybe more appropriate for lettuce, which is the other crop where we're getting a good fit besides carrot. So we align with the seed line and we inject the steam. And then the next step is planting. And you can go in and seed carrots, which we have done, or transplant in the case of lettuce or broccoli or another crop. So it really is a disinfestation method, and everything happens before planting. So after the steam heat passes, which is really only a few minutes, you can plant whatever crop you desire into that area. So this what it shows here is a 40-inch band that in a trial that we did here in Salinas. And in between those two white lines is the band, and where that was steam prior to seeding carrot, which is visible in the picture. And so what I call this is the product of this treatment. And that basically is a sanitized band area suitable for planting your desired crop. All that other green material there, they're weeds. We have a rather weedy site here in Salinas, and which we maintain, much as the nematologists maintain their nematode nurseries. We have weeds, and the nice thing about weeds is that they're highly visible, so you can see the results. So this is a photo of the machine. I'm showing you the results from the past and up in 22 and 23. That's a Cymox steam generator from France. Cymox is a French company, Eastern France, practically in Switzerland. And so what we found in over the past 2 years of using that machine is that we get excellent control of purslane, 99% on the average of a number of trials. Shepherd's purse, nettleleaf goosefoot, we get about 88% was the average. For burning nettle, handbit, and pigweed, we basically completely control that weed. Little mallow or cheeseweed, if you like, is a bit more difficult. I worked a lot with fumigants. That particular weed happened to be more difficult to control with fumigants as well. It could be a combination of it's a larger seed and ability to come up from deeper in the soil. It's also probably got some resistance to penetration. I know that's true for that weed and seed in the case of fumigants. So the data that we have collected, weed densities, or basically weed control, hand weeding time, to try to provide some economic data, which is hopefully more useful for growers in assessing the value of whether this treatment has potential for return on investment. We are assessing Pythium control because we are set up for that, and we assess crop yield. And the treatments that we assess that I'm going to talk about here are basically steam versus no steam. And these are replicated trials. And so here's some results in terms of the weed densities. And this is from a lettuce trial last year. And so basically that's showing the number of weeds per square foot. So the control there was almost 70 weeds per square foot. So quite a high density. And so what that shows is the 4W and 3 deep, so 4 inches wide by 3 inches deep. We want to assess, we want to know what the dimensions of the treatment area needs to be. And so there are some different characteristics in terms of weed control, what's optimal, and for disease control as to what's optimal. And so 4 inches wide, 5 inches deep, 5 inches wide, 3 inches deep, and 5 wide by 5 deep. So those were the treatments. And then the hand weeding times. So what we're seeing for weed control is the dimensions don't vary much. And so the hand weeding time are all about the same, regardless of the dimensions. So the steam is doing the trick. The dimensions are more important when we look at Pythium. And what we have are 3 columns here. We have the treatments of the first number is the 4 or 5 is the width of the band. And the second number, the 3D, is the depth, 3 inches deep or 5 inches deep. And so the Pythiums, we did soil samples before treatment and then after, a day after. And so there is some variability. This is a natural Pythium population, so there is variability there. But the important thing is to look at the before compared to the after. And what it shows is that the 5-inch deep treatment completely controlled the Pythium. Fusarium is also a huge problem in lettuce, and what this data are showing from a commercial farm is a significant reduction. So this is measured using the Kamada measurement, extremely important, challenging disease in lettuce. But this is what we're getting, a significant reduction in Fusarium. We get a lot of questions about what are we doing to the soil microbial community? Are we damaging it? And this is some new data we just got in the last couple of days. I have a graduate student, Erica Escalona, working on this. And what we have done is extract DNA from the soil samples before and after. And what this shows in the purplish color shows the pre-samples, the pre-steaming samples, and the green shows it after. And we've done this in strawberries a few times. So what obviously you get a reduction in the amount of DNA in the soil. And this is measured on the 16S RNA, which the soil microbiologists use as a standard method. And what they found is that there's a group of organisms that called the Firmicutes, which are promoted. And as I understand it, I'm not an expert in soil microbiology, but we are working with a number of people who are. And so the Firmicutes are apparently quite beneficial. I was very concerned about the nitrobacteria, and there's a group called the Nitrospirota. That's how they classify it. And the data that we have from a spinach trial, it doesn't show any effect at all. So we're very careful to not oversteam the soil, because when you oversteam the soil, you can kill the nitrobacteria. And that is not a good thing. And that in greenhouse situations, that's where some ammonium toxicity has resulted in historical terms. And so you definitely do not want to do that. The way we are applying steam is continuously moving. And so we have a great deal of control over the temperature, because it's very foolish to oversteam the soil, because you're increasing your cost and you're creating problems. And so it's very much a Goldilocks scenario, not too hot, not too cold. You want it to be hot enough to kill the pathogens and the weed seed, but not so hot that you do damage to the soil. And that's what we are doing. And so I'm going to show you what's new. This machine we just took possession of from the shop and brought it to Arizona because we had too many wet fields here. And I've got a good collaborator down there, Mark Siemens. But this machine was funded by the Propane Education and Research Council, PERC, which is basically a national commodity board for propane dealers. And so that I have a video of that in operation. So notice that the bed, basically as soon as the soil cools, it's ready to plant. So that is set up for spinach in Arizona, just as you can see to the right. But literally you can plant any crop you want. There's another shot. I think Okay, this is a different one. I just wanted to show the temperature. So that there are shanks under that plate that's injecting steam into the soil. And so what this shows is that's 200 degrees Fahrenheit. We need to go faster because that's too hot. So the objectives that I am proposing for in Carrot, based on the conversations that we had a couple of weeks ago, we're changing our design. It sounds like the emphasis, the request of the industry is basically to control the weeds, and so we are redesigning the injectors. And so what this shows is what we're looking at for carrots. The light green, the lime green plate in the top is, we call it the top plate, and that is through which the shanks are inserted. So this is all laser cut steel, and so we have to get it right before we put the request into the shop for cutting with their laser, and it does a really nice job. Amazing. And then so the bottom shows kind of some of the front view of the shaper where it's pulling the soil into the bed and shanking in the steam. The difference between this to what we're working on still as of yesterday and today is we're going to respond to the need to control the weeds across the entire bed top. So this design already has been changed, but that's what we're doing. So that is my last slide.
Thank you, Steve. And we have a couple of questions in the Q&A.
Yeah. Sounds very energy intensive for a crop that retails at low cost. Well, I think the point is, what is the cost of production? Are we reducing the cost of production? I mean, I'm hearing quite high expenses for hand weeding and organic carrot. And if we can reduce that significantly, that's the objective here. How effective is steam versus fumigation? Are there some weeds that are triggered by the steam treatment? I'm not aware of any weeds that are triggered. We've compared steam in the strawberry nurseries side by side with methyl bromide, and it is the best fumigant, and we were on par with it. And strawberry plants are extremely sensitive to disease. So yeah, are they triggered? I mean, that's been talked about with fumigants. They're stimulating the weeds. There's actually quite a few weeds like clover that build up in fumigated fields. That's a problem in strawberries. And I also know a problem in a flower field that has been repeatedly methyl bromide fumigated, where you build up a population of clover. It's not so much that they're stimulated as that they survive. Okay, yeah, finish bed. Thank you. I give credit to the engineers I'm working with. Time frame to follow the steamer with a planter as soon as it cools. You can steam in the morning and plant it in the afternoon. This is what I tell growers. It doesn't sound very scientific, but it is. Put your finger in the soil, and if it's cool, if it's not too hot, you're ready to go. You can put drip tape in. You can put you can plant whatever you desire. You don't want to go immediately afterwards to plant a seed, because you'll do the same thing you do to the weeds. You kill them. You need to wait till it cools, but it literally does. You don't need to wait long. Will this machine have long-term control on the weed seed bank? It basically is only going to reduce the seed bank in the area that's treated. So it depends on how big of an area you treat, but the effect, as soon as you go into the field and do some deep tillage, you're going to bring up weed seed, the seed bank from deeper in the soil. But the seeds that are dead for forever. But it's expensive to treat the whole thing.
Maria, could you please launch the poll?
Well, the first question is a true or false question, and it says an update on the state of deficiency of weed and disease control. And then the second question reads, steam treatment of soil is either pasteurization, sterilization, or none of the above. Most people put true, but the answer is actually false. And most people put sterilization for the second one, but the answer was pasteurization.
Can I comment on the second one?
Yes.
So, yeah, the correct answer to that one is pasteurization. Maybe I didn't emphasize that enough, but that's why I and maybe it's not a good analogy, but I use the Goldilocks, not too hot, not too cold. And so pasteurization is something that, I think it's most familiar with people as far as milk. You do not want to cook milk. It takes on a different consistency if you cook it. So you pasteurize it to remove the pathogens, or if there are any in the milk. And what we're doing here is we're getting warm enough, hot enough to kill the pests, but not do lasting damage to the soil microbial community. And this has been looked at in with steam and with fumigants, and there is a happy medium. And the soil microbial community the beneficials are amazingly resilient, and that's how we maintain fertility in these situations. So,
all right, thanks, Steve. We are running behind our schedule, so we should jump on to our next presentation by Dr. Jeremiah Dunn from Oregon State University, and he will be providing an update on SCRE-funded bacterial blight research. Jeremiah?
Yeah, thanks for the introduction. Today I'll be talking about an update from some of our SCRI-funded research, focusing specifically on some epidemiological questions that we've had in carrot seed production systems in Central Oregon. So a little background on carrot seed production in Central Oregon. This area is known to produce anywhere between 40 to 60% of the national and world supply of carrot seed annually. And amazingly, that's done on anywhere between 2,000 to upwards of 5,000 acres of carrot seed. It's a pretty labor or a pretty intensive crop to grow. It's drip irrigated and bee pollinated. And there's two primary methods of carrot seed production. The first is a steckling to seed production, where roots are grown in California or Arizona, and then they're transplanted. They're dug up in the winter, transplanted in the spring here in Central Oregon, and then they're harvested in the fall of the same year. The other method is seed to seed production, and in this scenario, hybrid lines are planted in August, allowed to vernalize in Central Oregon, and then harvested in September of the following year. So in the seed to seed production cycle, there's actually overlapping production cycles where the seed that's being harvested is actually harvested after the seed crop for the next year is planted. So there's a little bit of overlap there, and that's important when it comes to what I'll be talking about later. Now, bacterial blight of carrot is one of the more important diseases impacting carrot seed production. It's caused by the bacterium Xanthomonas hortorum and the specific pathovar of that species, carotay. It was first reported in California in the early 1930s, but it's really found wherever carrots and carrot seed is grown. The issue with this pathogen and this disease is that it affects leaves, petioles, umbels, and seed. And the impacts on both the plant and the umbels and seed are important in Central Oregon. Some of the first symptoms you'll often see on leaves are these yellow angular spots that often occur here at the tips of the leaves. And then they will expand into some irregular brown and water-soaked lesions And that's one way you can tell them apart from maybe some of the fungal foliar leaf pathogens that you might experience in carrot root production. Now, this pathogen is both seed-borne and airborne. And the interesting thing about this pathogen is that it can survive and reproduce epiphytically, meaning it can be found on asymptomatic plants. And really, symptoms don't start to develop until we tend to see large populations on the foliage, approaching about 1 million bacteria per gram of leaf tissue. And so this figure here is showing some populations of Xanthomonas on carrot seed crops going from about mid-June to mid-July. And you can see in most cases by mid to late July, most of the carrot seed crops that we were looking at in this case had relatively high populations above that that 10 to the 6 level that's indicated here with the red line. And so these foliar populations can get very large within a carrot seed production season, and they can cause symptoms, or they can be asymptomatic in some cases. Now, there's several negative impacts of bacterial blight on carrot seed production. For the seed growers in our area, there's costs associated with management, typically using copper bactericides. But then there can also be reduced yield, both due to compromised foliage that is diseased, or particularly when the disease impacts umbels. As you can see here with the middle picture, this flower is blighted, and you can imagine that seed yield will be reduced in that case. In some instances, when the disease impacts umbels early in their development, the bottom right picture is showing an umbel that was essentially killed before it could really fully develop. And so those are the issues that we face here in the seed production side. And then on top of that, for the seed companies, they have to oftentimes utilize expensive and difficult hot water or chemical treatments to seed lots. And in some cases, seed lots with high or any levels of Xanthomonas might not be accepted in certain markets. So in terms of management of bacterial blight in carrot seed production, we can kind of have 3 main strategies or 3 main areas where we can do this. We can do it in the field. And so we can do that by those foliar bactericide applications, as I mentioned. Coppers and coppers plus mancozeb are often most commonly used. We can avoid overhead irrigation, and that's often done in about 90 to 95% of our seed crops, which are drip irrigated. And then also growers can incorporate those infested seed crop residues. There's quite a bit of carrot seed crop residue and debris that's left over after combine harvesting. And so if that can be burned or buried, that can reduce pathogen populations in the residue. I mentioned those hot water and chemical seed treatments that can be applied post-harvest. Those are often very tricky to perform, especially on large lots of seed. And then there's also issues with disposal of the effluent that comes after the treatment. Finally, and probably the most effective, is excluding the pathogen completely from production. That can be done through pathogen-free seed or stecklings. And then also in this area, we often have a 2 to 3 year rotation, oftentimes even more. And that provides some spatial and temporal isolation between fields. However, in most cases, we'll have certain isolation pockets where carrot seed of the same or similar varieties are being grown. And so in those certain isolation pockets, we might actually have potential for spread between and within fields. And that's kind of getting to the crux of our question here. What's the potential of the Greenbridge effect, and how can we assess risk? I mentioned that large epiphytic populations of the pathogen can occur on carrot plants. And that 13-month production season in the seed to seed production scheme results in an overlap of about 1 to 2 months between carrot seed cropping seasons. And so that provides a potential green bridge effect starting when the crop is harvested, and that airborne debris could potentially move onto new fields. And Lindsay Dutoy did some work on this many years ago, where she was able to detect airborne Xanthomonas up to about a mile from crops that were being currently threshed. So this is an example of what we often see in September in Central Oregon, carrot seed piles here on the ground, swath piles, and the combine coming through and harvesting. And in these high desert conditions, we often have quite a bit of dust resulting from the crop debris, and that can get airborne quite readily. So we've done some work to look at the potential for XHC dispersal or Xanthomonas dispersal within and between fields. And we deployed some passive traps at different distances from combines that were actively harvesting commercial carrot seed crops. And as you can see, we were able to detect the pathogen as measured by the genomes per filter or per trap, anywhere between 12 to up to 156 feet away from the combine, suggesting that these plumes that contain Xanthomonas are likely spreading within the field and onto potentially other swath piles, but also maybe between fields, especially those fields that are located close by within an isolation distance. We also looked at the effective particle size on the amount of Xanthomonas that we detected. So we tracked particles that were less than 50 microns, all the way up to particles that were above 400 microns. And while we tended to trap particles, the large 400 and larger micron particles closer to the combine, we were able to detect particles, small and mid-sized particles, quite a distance from the combine, and we were able to detect anywhere between less than or about 10 colony forming units per particle or per trap, all the way up to maybe 100 colony forming units. And it's really these small particles, I think, that could pose the most risk for long distance dispersal between and within carrot seed fields. Now, there's not only the potential for Xanthomonas to be spread onto other carrot seed crops during combining events, but being a particularly good epiphyte, Xanthomonas species can occur as asymptomatic non-hosts. And so in 2022, this is an example of some of the other crops that we grow in Central Oregon: grass seed, alfalfa, wheat, and different forages, as well as parsley seed and peppermint. And we've done some sampling previously that demonstrated that we could detect viable Xanthomonas on some weeds and non-carrot crops in Central Oregon. So what we weren't quite sure of based on how we sampled and when we sampled was whether or not these were transient or resident populations, and whether or not these can serve as potential inoculum reservoirs in carrot seed production systems. So I had a graduate student who tried to address this question, and she went into 3 different farms and sampled foliage from these farms from both the carrot seed crop and then also any of the fields that were surrounding that carrot seed crop. So in Farm A, she sampled carrot seed crops, parsley root seed crops, and some alfalfa. Field B or Farm B, we sampled carrot seed, parsley root seed, and Kentucky bluegrass seed crops. And then in Farm C, we sampled carrot seed, parsley, and forage rye. And as you can see in the middle column, in all cases, we detected the most Xanthomonas on the carrot seed crop foliage. However, we were able to detect very similar, in some cases, amounts of the pathogen on asymptomatic foliage. For instance, in Farm A, we found it on both parsley and alfalfa. Farm B, we found it again in parsley and on the Kentucky bluegrass seed crop. And then on Farm C, where we had the highest levels, we had very significant amounts on parsley seed and even on forage rye. So Kate went on to do some replicated experiments in the greenhouse where she planted some example rotation crops in the greenhouse, including carrot and alfalfa and some of the other ones that we looked at in commercial settings. And then she inoculated them and then tracked the population dynamics over time. And so she inoculated them, sampled them the day after, as indicated by the black bar here, and then at 7 days, 14 and 28 days. And so these are showing the mean log genomes of the pathogen that we detected with PCR on the different foliage types, and the error bars are just representing the standard deviations. And so as you can see on carrot, she started off with about 10 to the 5th bacteria per gram of leaf tissue. And by the end of the experiment at 28 days, she was detecting almost 10 to the 8th bacteria per gram of leaf tissue. Alfalfa, on the other hand, we tended to see a decline in populations over that 28-day period, going from a less than 10 to the 4th down to about 10 to the 1st bacteria. However, some of these other crops did show relative increases in the population numbers over time, including curly parsley seed crops, Kentucky bluegrass and rough-stalk bluegrass, and even winter wheat, suggesting that maybe these crops might serve as potential viable reservoirs for the pathogen. So in conclusion, what we've shown is that the Xanthomonas pathogen can be detected in airborne carrot seed crop debris that's generated during harvest. We were detecting it up to 150 feet away from the harvesting activities. And we also detected it on the very small particles, which can potentially travel long distances. Finally, the work done by Kate in my lab demonstrated that non-carrot crops can potentially harbor asymptomatic Xanthomonas, even at high levels, and may serve as potential reservoirs of inoculum in the carrot seed production systems of Central Oregon. So with that, I'd like to thank Kate Baldino, the student in my lab who worked on the greenhouse and alternative host experiments. Matt Huckins is a PhD student with the University of Utah who helped us out with the airborne trapping, our grower cooperators, and then of course our funding from the USDA Specialty Crops Research Initiative. And with that, I'd be happy to take any questions if there are any.
Thank you, Jeremiah. And we have one question in the Q&A.
So the question, to what extent do you think copper fungicides help existing Xanthomonas population problems in commercial fields? And do you think that repetitive use of copper fungicides stress carrots and make them more susceptible to other diseases like alternaria? Also, is the efficacy of copper fungicide on Xanthomonas dose dependent? Those are some really good questions. I think that in the research that we've done here in Central Oregon, copper fungicides can reduce the population on carrot foliage by upwards of 2 log, so 10 to the 2 reductions. But what we tend to see is that those populations rebound back within a certain period of time. And so, you know, even on a 14 or 28-day rotation of fungicide of copper products, we often see that those populations tend to rebound. In terms of the repetitive use of those coppers and the potential for stress, I'm not sure about that. We don't necessarily see alternaria much in this production region. Powdery mildew tends to be more predominant. And that tends to come in later in the season as the crops are maturing, and also during the period of time when bees are out and we're not able to perform any control measures. And the efficacy of copper is dose dependent. To be honest, we've only looked at some of the labeled higher rated doses, and so we haven't looked at lower dosages in the research I've done.
Thank you, Jeremiah. And Maria, do we have poll questions? Yes.
The bacterial blight pathogen Xanthomonas can potentially be spread throughout seeds, seconds, and airborne crop debris. The answer was true. And for the second one, true or false, spraying crop debris is a potential management practice for bacterial blight in seed crops. The answer is also true.
All right, thanks, Maria. And now it's time for our guest speaker, Kenneth Miller. He's a soil scientist and agronomist with Formation Environmental LLC, and he will be presenting on managing nitrogen and irrigation for sustainable crop production.
Thanks, Jaspreet. Just pull this up here. All right, thanks. Good morning, everybody, or almost afternoon here. Lots of interesting topics covered today, and my hope is to provide an update and inform a little bit on some considerations around managing nitrogen and irrigation and carrot production systems. As Jaspreet said, I'm Ken Miller, working on behalf of the Southern San Joaquin Valley Management Practices Evaluation Program, which is a required program under the Irrigated Lands Regulatory Program. And within our boundary, we have quite a bit of carrot acreage down in Kern County area. And so was invited here to talk about some specifics related to the Central Valley, and then also more broadly about general nitrogen and irrigation management. And so today I'm going to focus on a specific requirement under the Central Valley Irrigated Lands Program. So there are different regional boards around California that have different implementations of their irrigated lands programs. And so this is specific to the Central Valley, the things I'll be talking about. And that's specifically called the groundwater protection formula and values and targets. I'll define those, and then we'll talk more broadly about inefficiency in carrots. And so just a brief overview of the participating coalitions that have engaged on this groundwater protection process in the Central Valley. There are 13 of them that are involved, highlighted here. The one commodity or the one coalition that's not involved with this process is the California Rice Commission, because of the nature of nitrogen cycling in that cropping system and just the extremely low risk of groundwater contamination. Rice was exempt from these requirements, but all the rest of the Central Valley watershed, as outlined in blue here, is engaged in sort of a unified approach to representing the crop root zone and impacts to water quality, including carrot systems, as well as all of the other systems, except rice. And just for a quick reference, the SSJV MPEP is basically functioning for coalition 7, 9, 10, 11, 12, 13, and 14. So the southern portion of the Central Valley. So real briefly, what is the groundwater protection process? It's made up of 3 pieces, as defined in the orders. And so there's what's called the groundwater protection formula, which is basically a means of estimating how much nitrate is discharging below the root zone from irrigated agriculture. And so that's what the value represents, is an estimate of that nitrate leaching. And the orders require these estimates to be aggregated at a township scale. And so they are not reported on a field by field basis, or not even on a crop by crop basis, but aggregated across all crops and all fields in the irrigated lands program within a township. And then ultimately there's what's called the groundwater protection target. And the purpose of the target is for setting a goal, a target for that township to ensure that the amount of nitrate that they're discharging does not cause or contribute to an exceedance of the water quality objective in the future. And so it's not really about remediating the past. It's about making sure that irrigated agriculture today is in compliance moving forward. And so these targets were submitted back in late 2022 and were relatively recently approved in June of 2023. And they are to be revised and updated on a 5-year basis. And so we have sort of our first set of targets and are committed to refining our modeling framework and our tools to estimate them over time. But we have put out a first version, and they have been accepted. And so I'll talk a little bit about each of these pieces in more detail so that folks can get a sense of how all of this works and how it affects growers and how it ties into considerations of nitrogen and irrigation management. So just real quick, what are the townships that are affected by this process? As you can see, it's the vast majority of the Central Valley floor that's the townships currently involved. There's 399 of them outlined in blue, and they're defined based on high vulnerability area, which the coalitions were required to determine through groundwater assessment reports. And so all that red area is determined to be areas on the valley floor that are vulnerable to groundwater contamination of nitrate. And so if the township has sufficient high vulnerability area within it, then we have to compute a value and a target. And so you can see that, quite a bit of the valley is subject to this, if not most of it. So to characterize the root zone, what the coalitions have embarked on is a multi-year process to come up and develop what we're calling the Central Valley Soil and Water Assessment Tool, so CV SWAT. And so SWAT is actually from the USDA. It's been around a long time and been refined for a long time and used widely within the scientific community to do all sorts of environmental modeling assessments, nitrate, phosphorus, sediments, pesticides, etc. And the coalitions opted to use this tool to characterize the root zone of irrigated agriculture because it had a lot of things going for it. We know that field specificity is important and that the nitrogen cycle will play out in different ways depending on local conditions, right? And so SWAT allows us to represent the root zone sort of at the subfield scale across the entire Central Valley. We're running it at a daily time step, and so feeding in CIMIS data on a daily basis, programming management decisions on a daily basis, it's including soil information from the NRCS. It has an active crop growth model, and it's comprehensively representing both the water and the nitrogen cycle. And so it's doing a lot for us and trying to give us as accurate a representation as we can develop at the scale we're trying to function on, which is the Central Valley, spanning about 6 million acres more or less of irrigated agriculture. So quite a big extent that we're trying to characterize here. And so the coalitions adapted SWAT. This work started back in 2017, I believe. So it's been going for a while. There was support from the NRCS. They awarded the SSJV MPEP with a $2 million conservation innovation grant. And so part of that funding, as well as funding from the coalitions, helped develop CV SWAT. At this point, we've got over 50 calibrated crop models that represent the vast majority of the irrigated acreage. That includes carrots. And we've basically worked with industry experts, farm advisors, et cetera, to basically detail out representative management schemes, and also, of course, utilizing grower reported information that's collected through the ILRP to ultimately spin up as accurate and representative of a process as we can. And so on the right, we just have a map showing some of the detail in CV SWAT, highlighting all that. And I actually have a zoom in here for the southern portion of the San Joaquin Valley, and just highlighting here townships that have some amount of carrot acreage within them. So you can just kind of put that into context. And so each of these different colored polygons is an actual modeling unit in SWAT, and it's reflective of the soil mapping units. And so you can get a good idea of the amount of detail we're able to represent. And so ultimately what we ended up doing after we calibrated SWAT on a baseline basis was we did many simulations of it, over 75,000, to try to represent all of the different scenarios that growers have reported in terms of nitrogen management and yields that they realize. And so in the case of carrots, we have, a little over 550 different combinations of nitrogen applied and yield that are simulated within each one of these units as sort of like a database. And so the way that database actually gets utilized and a value for a township is calculated is if you just take this example township here, which is 6 square miles by 6 square miles, and if we just focus on an individual parcel, we can see that in this case, there's actually 6 unique soil mapping units associated with that parcel. And so because we know this APN, the APN of this parcel, we can tie that back to an individual irrigation and nitrogen management plan, which growers are submitting on an annual basis, right? And those plans tell us, one, the parcel, so we know where it is in space, but they also tell us the crop that's grown. They tell us the nitrogen applied by the grower from all the different forms, and they tell us the yield. And so we know a lot of information about, what's going on here, a lot of important information. And so the way the process works is that we actually go into that big database of CV SWOT output. And for each of these different soil units, we're going to have a run that is either is or is very close to this nitrogen application rate and this yield. And so that simulation will have an estimate of nitrate leaching with it, right? And so we're actually doing that for each field or each parcel within a township. So all these black outlined areas, we do this process for each of them and then roll all that up to create one number for a specific township in terms of leaching below the root zone. And so I emphasize this because, I think sometimes growers and advisors wonder, how all this data being collected is actually utilized. And this is an important mechanism here. And so really the way in which we're trying to characterize and understand, the nitrogen dynamics within the Central Valley context is relying on grower reported information and feeding that through a process-based model to come up with these estimates. And so the INP reporting is very much important and is utilized regularly in this assessment as well as others. And so there's definitely an important end use. And so what you can see is those same townships that have some carrot acreage. I'm just showing what the township values are. And these are averaged across all the crops and all the acres. Again, we just have to report one aggregated number. So we're not exposing individual growers nor individual crops. And this histogram is just showing that, the percent of the INP acreage that is in carrot varies, but typically it's not a majority acreage in any of these particular townships. So it's ranging from anywhere from, less than 1% up to almost 15% in one case. But just to give you a general sense of, each of these townships has a different complexion, but these ones have some amount of carrots within them. And so that's kind of just the general workflow for creating nitrate and loading estimates below the root zone, and the coalitions have done that and will continue to do that moving forward. And so in terms of groundwater protection targets, where do we need to get to? What is protective of water quality? I only have a half an hour today. I could spend, 2 days talking about all this stuff, and I know I'm going quickly, but I want to get to the rest of the presentation. And this is really just kind of the setup to why we, care about nitrogen efficiency, or one reason to care about nitrogen efficiency and irrigation efficiency. But when we talk about groundwater protection targets, what we're really trying to do then is take these values, right? These loading estimates from irrigated ag, but connect them to all the other processes within the Central Valley that ultimately affect water quality, right? So it's not just irrigated ag that is the only thing affecting water quality, right? We need to understand what the hydrologic conditions are, sources of recharge, et cetera. We need to understand what else is impacting the aquifer and water quality. So it's not just irrigated ag. To the degree that there's attenuation processes happening below the root zone, we want to account for those, give credit for them. And then ultimately, understanding what is happening within the aquifer and how does that, manifest into water coming out of a well, which is the water that we actually use, right? And so with nitrate issues, we're really focused on drinking water, right? There's a human health risk associated with elevated concentrations of nitrate. And so, looking at produced water in domestic, supply wells and municipal supply wells is important to us. That's the thing we're trying to understand. And so we actually do have a groundwater model that we're able to use. It's called the Non-Point Source Assessment Tool, NPSAT, and that was developed by researchers at the University of Davis. At this point, over a decade of work has gone into developing that model, and we work very closely with them to evaluate it and to continue to refine it. And again, I just don't have enough time to cover all these details, but just know that we're using a groundwater model from UC Davis that allows us to consider all of these processes and gives us answers in virtual simulated wells. So in terms of what the coalition submitted to the regional board, there's actually two different metrics that we've put forward. We've put forward what we are calling groundwater protection milestones. And these were not required, but it basically put them forward as a near-term load reduction goals, based on an analysis of INMP data and trying to identify, where within these townships do we think that there's, some low-hanging fruit that would be, that could be prioritized to lower the overall loading from the township without, substantial, hopefully, or very difficult transitions. Just some outreach and education would hopefully help, handle that. And so we did put forward these milestones as well as the targets themselves. And so the targets are what are required, and they're basically identifying whether or not the township will be in compliance moving forward. And again, we use the groundwater model for that. And so I have maps of each of these for these same townships, just to illustrate, from a qualitative standpoint that, the milestones basically, help us along the way, right? And they don't, they're not necessarily protective of water quality, but they do sort of demonstrate that just through implementation of some BMPs, best management practices related to nitrogen and irrigation management across cropping systems, we can achieve a substantial amount of load reduction and get us, on the path of where we need to get to with hopefully the least amount of difficulty for that step anyways. And then when we talk about targets, unfortunately, those are shaken out to be, more difficult than the milestones in many cases. And again, I say that these are sort of our first draft, not draft, but our first version of the targets, and they are to be updated on a 5-year basis. And so we will be continuing to refine our modeling, our data inputs. There's a lot of complementary processes going on that it will help us do that. But this is sort of our first cut and where we need to start working towards. And so just the reality is that, load reductions will be needed in many portions of the Central Valley, including townships that have carrot acreage. And so carrots, being a piece of the puzzle, can help, contribute to the township's ultimate goal in terms of how much nitrate is loading below the root zone. And so just a couple bullet points, some clarifications before I get into the nitrogen efficiency stuff. At this point, these interim milestones and the targets, they're not technically enforceable metrics. But, at any point, the state water board, which is above the regional board, they in the future, they may consider, what are appropriate enforceable metrics and impose them. At this point, we're not there. And, truthfully, to the degree that we can show, we've done a lot of effort to try to characterize this issue and set targets. And if we can show progress towards meeting them, then that's great, and potentially helps us, remain in sort of in a position we're in now where we have to do reporting, we have to come up with these metrics, but at least we're maintaining flexibility, we're maintaining the township's ability to decide how to manage and achieve these targets. And that's sort of the best case scenario we could hope for. And so progress will help maintain that situation, hopefully. And just to clarify, how does this all shake out and get communicated? And how does it affect, growers or their advisors? So coalitions are required to develop what are called groundwater quality management plans. And so within these plans, they have to include specific schedules and milestones and performance goals. And so within these plans, the groundwater protection targets will be incorporated this year. So the coalitions will be saying, okay, here's our general plan to try to work towards meeting these targets. And so what that really means is that each coalition is going to really have their own individual strategy and approach for prioritizing outreach and education to growers and advisors. And so that'll shake out differently depending where you are in space, but it will be a process where the coalitions help folks understand, what's in front of them and provide some resources for how to get there. But that's still kind of evolving and more will be coming within the relatively near future. But just some key things to think about when we're talking about all this groundwater protection stuff. And then so, here's this issue, right? Here's this regulatory requirement. And, how is it that we get there collectively? How do carrots, help get there collectively? And so just wanted to talk a little bit about some basic analyses that we've done specific to carrots, and then highlight some additional resources and upcoming trainings for sort of deeper dives into all of these topics. But so, when we think about nitrogen efficiency, if you recall those irrigation and nitrogen management plans, which I'm showing here on the bottom, the stuff that's, the different attributes that are reported, with these plans, we can get a basic nitrogen balance, right? And so when we say nitrogen balance, in this context, we're talking about a simple N balance, which is basically you have your inputs, right? Your nitrogen application, which is all these different sources. And then you have yield, and there's an estimation of how much nitrogen is removed with that yield. Right now, the estimate is about 2.8 pounds of nitrogen per ton of carrot. And so if we have input and we have output, we can solve an N balance, right? And so in theory, the bigger that N balance, the greater the risk there is to leaching. I have another slide to talk about the fact that N balance does not equal leaching. And that's why we use the SWOT model to give us a better estimate of what may actually be happening in terms of leaching. But it is an indicator, right? And as the N balance goes up, our risk of leaching also goes up. And so again, this is just highlighting sort of the importance of the INP summary report, and really, to have something that's implementable at the Central Valley scale and providing relatively useful information, especially when you're looking over space and time, it's kind of an elegant reporting mechanism. And so just a real quick reminder, I think folks have seen this before probably, but where does that nitrogen removal factor come from? So we have been working with researchers at the University of California, Dr. Daniel Geisler specifically, to implement two FREP-funded projects, CDFA Fertilizer Research and Education Program projects, to sample crops, including carrots, and analyze them for their nitrogen content to get, California and Central Valley specific estimates of these N removal rates. And so basically you can see over about 4 years, we collected samples from over 60 different production areas and analyzed them for their nitrogen content. And so the average shakes out to about 2.8 pounds of nitrogen per ton. Of course, there's a range, there's variability, but we're taking an average here, and when you look across a big unit of area, it should tend to sort of work out. And so it might not be 100% perfect in one field, but when you kind of look across space, it's a reasonable estimation. So that's where that number comes from. When we actually look at what growers are reporting, it's kind of some interesting findings. This is just showing some histograms of different N application rates, and the fraction of reported acres that are falling within each of them. And you can see that there's generally, it's not like a normal distribution. That's kind of spread out across a relatively wide range. And if we look across just 3 years, we don't see on average big changes in N rates, and we don't really see on average big changes in yields. And so, and not to say that you would expect huge changes from one year to the next, but just showing that we're generally not seeing much in terms of changes over the last 3 years, anyways. And of course, down in parts of the Central Valley, organic production makes up a substantial acreage, and so I don't have the numbers shown here, but we did look at organic versus conventional, and interestingly, the yields are relatively comparable, but there are some differences in average N rates, and I'll talk a little bit more on that on subsequent slides in terms of organic nitrogen management considerations and the challenges associated with that. But we do see some differences there. But this is just kind of giving you a general snapshot of what we're seeing on average over time. And, going and studying agronomy in school, and, we all learn about the idealized relationship between fertilizer inputs and yields. Economists love those. But then when we actually look at INP data, we get these shotgun blasts, right? And nitrogen application rate alone is not a very strong predictor of yields in these INP reports, and we really see this pretty much in most other crops, if not virtually all of them. There's just other factors that are not captured within the INP that are affecting yield beyond just N application alone. But this lack of relationship that we observe, what that ends up meaning from an N efficiency standpoint is that we do really tend to see, as you apply more nitrogen, the A-R N balance does go up, right? And if you look at these correlations, they're very tight. And if you look at the slopes, it's around 1, right? And so basically, the N balance is increasing linearly and basically at the same rate as increases in N application rates. And so, N balances for carrots, tend to be higher than some of the perennial crops, but sort of fall in the middle of the pack of annual crops in terms of what is typical. But I think this relationship is important to understand. And as we think about, achieving targets, and trying to minimize nitrogen balances, there is just that basic principle that as you're putting more on, you're probably contributing more to an N balance. And so that's just something that we want to highlight and emphasize, as people are developing and implementing nutrient management plans. And so, as I mentioned before, nitrogen balance does not equal nitrate leaching, right? We go to the process-based swap model to actually simulate where the nitrogen is going, right? So we know there's gaseous losses through denitrification or ammonia volatilization. There can be some storage in soil organic matter, some turnover. But the reality is that particularly in most annual cropping systems, these other pathways are a relatively small piece of the pie, right? So talking about 10% less or so of applied nitrogen. And so while we do acknowledge them and reflect them in our actual leaching estimations, the reality is that they're not alone going to solve the issue of nitrate loading to groundwater. And of course, there are other factors that are more nuanced than what is fully captured in a single year in an INP. We understand that there's effects of rotations and cover cropping. The important caveat there is that if you're using a cover crop and scavenging some nitrogen, you need to be taking account of that scavenged nitrogen, and it's going to be available, or most of it's going to be available next year. And so if that's not really accounted for, then the benefit of the cover crop from a water quality standpoint is not really realized. And so while we do have these nuances, there's caveats to them. And so, as we look at INP data year over year over year, these effects more or less get captured, right? Because if folks are not adjusting their fertilizer inputs as a function of residual nitrate from rotations or turnover from cover crops, if that's not really reflected in the applied nitrogen, then it's kind of a wash. And so that's an important consideration there. So if you are cognizant of these factors, just make sure you're keeping track of them on the back end as well and adjusting accordingly. And last but not least, as sort of the schematic for targets implied, there are potential processes below the root zone. So once nitrate is left the root zone for attenuation, and that's really, it depends on where you are in space and what those conditions are. And it's not necessarily ubiquitous, but it can be important in certain areas. And so we are keeping track of that in the groundwater protection framework. So, what's the way forward, right? Understanding where we're at and where we're trying to go. The simple fact is, in areas where we need to achieve load reduction, it boils down to this simple mass balance, right? Where we need to be lowering this A-R number as best we can. And the way we do that, or the result of implementation of practices, leads us to either put on a little bit less fertilizer, get a little bit more yields for the amount of fertilizer we're putting on, or some combination of both, right? But in essence, the A needs to get smaller, the R needs to get bigger, or both those things need to happen. And when we look at sort of the distribution of realized, A-R values for carrot growers, we see there's a wide spread, right? And while we have a median of about 80 pounds per acre, the tails can go substantially higher than that, right? And so, if understanding what are our opportunities and how do we help folks, towards the right end of the graph, get towards the left end of the graph is really just kind of the name of the game. And a big portion of that is just understanding how much nitrogen you're putting on relative to what the crop can take up and utilize. And understanding that in general, the more nitrogen you put on, the higher your nitrogen balance is going to be. And so that basic principle, kind of feeds in to all of this. And so the question is, how do you confidently reduce fertilizer inputs or increase your yields or both? And obviously that's a case by case and a nuanced thing to deal with. But in general terms, we like to advocate the process of planning, developing a nutrient management plan. What are you hoping to do? Monitor, look at soil samples, look at crop growth, keep track of what's going on. And, as you're monitoring your system, you're adapting, right, as needed, to inform the rest of your growing season. And so this is sort of the basic playbook, if you will, for helping to achieve these goals. We know that irrigation efficiency is absolutely critical for good nitrogen use efficiency. So if you're not necessarily highly efficient with your irrigation, nitrate is very mobile, right? And it's going to be moving with the water that's percolating through the root zone. And so, if water is being put on beyond crop need, or if there's a really low distribution uniformity and you're putting on more water, then your likelihood of a low NUE is increasing, right? And so just understanding that these two pieces are just so fundamentally connected is a really major piece of the pie. And so to the extent that you can evaluate and improve distribution uniformity, to the extent that you can schedule irrigations based on information related to climate, information related to your soil, crop water status, keeping track of those things and trying to irrigate more consistent with crop demand, that's going to really help quite a bit with respect to achieving an optimal nitrogen use efficiency. Realizing that there are, a considerable fraction of organic acres in carrot, we figured just highlight a couple considerations, realizing that organic systems are particularly challenging when it comes to managing nitrogen. And so just to that same point about irrigation, really important time of year to be careful with irrigation is early in the season in organic systems, because at the beginning of the season, you may very well have, accumulations of nitrate, as your amendments or your residues are breaking down and mineralizing and making nitrate available. So there might be a big reservoir in the beginning, but obviously, crop root zones are very small at that time. And so as you're irrigating, if you're irrigating too heavily and moving that nitrate beyond access of the plant, it's difficult to, it may or may not be captured later as the roots get deeper. But so just trying to keep that early season nitrate in as high up in the root zone as you can early on is really an important thing to be aware of. Another really challenging but critical piece is trying to come up with an estimate of how much nitrogen is actually released from organic amendments. And so, what's the total magnitude? When is that release happening within the growing season relative to when the crop, is needing or taking up nitrogen? And so quite a bit of work's been done by Patricia Ledziski and Daniel Geisler, and earlier by Tim Hartz and others, to look at, what sort of release patterns of nitrate or of nitrogen could we expect from organic amendments. And so the two basic ingredients that help us get an idea is looking at the carbon to nitrogen ratio. And so if we look at the fraction of mineral N that is coming out of all of the N put in, the lower that C to N, the more N is released, right? And as we increase the C to N, less and less of that nitrogen is actually made available, and you can actually get some immobilization and it goes negative. And so just being aware of, we're trying to have a reasonable estimate of that C to N should be very helpful for just getting a general idea of how much nitrogen might release. The other key piece is just the total nitrogen in the amendment itself, right? And so it's kind of connected to that, but somewhat independent. And so as you have higher percent nitrogen in your amendment, you tend to see more of that mineralize and become available for crops. And so there's a calculator based on this work that's available at Daniel Geisler's website. I have the link included here. And this will help give you an idea. And it's including things like soil temperature and the different properties that you can input, C to N ratio, percent N, the amount you're putting on and when, and it'll use CIMMYS data and create an estimate. But Daniel and others would be the first to tell you that, these are models and these are estimates. But really the best approach is to start here, but really develop or implement a soil sampling program so that you're actually keeping track to the extent you can and understanding what sort of release patterns are you seeing in the field, how are your crops responding, and it might take a couple seasons, but you get a good sense of how these practices actually shake out. And obviously there's a lot of moving pieces here and a lot of complexity, so this is definitely difficult, but really important to that question around, how do we dial in that applied nitrogen term for these systems. And so again, that's the resources here for that. Another important thing, just trusting the fact that nitrate in your irrigation water is no different than nitrate in your fertilizer. It's equally as effective. And so if you're able to, you gotta know how much nitrate is in your well water, and then, understanding how much water you're putting on and how much the crop is actually taking up can help you calculate that credit. But just realizing that it can be a substantial amount of nitrogen in your budget depending on how much water you're applying and the concentration of that nitrate. And so just being aware of that, and plenty of work's gone in by Michael Kahn, Tim Hartz, and others to demonstrate that the N uptake efficiency from nitrate in irrigation water is just as good as that in fertilizer. And so just there's no difference there. And so give credit for that as you can. And then, there's all these moving pieces, there's all these factors. How do you keep it all straight? Sometimes it can be very helpful to utilize a decision support tool to kind of pull all this information together, keep track, and make informed decisions within the actual growing season about when to irrigate and fertilize and by how much. And so there's, of course, many different options out there, and I'm sure many of them are good. The one I was just going to talk about today is called Crop Manage. For those of you who haven't heard of it, it's from University of California, A&R. So it's free, which is nice. It doesn't require any special extra equipment to be purchased. It's designed to be usable in the field, and so it's relatively easy to use and keep track of your data and manage your data. It's integrating soil information, climate information, crop, as you tell it what you've done in the past in terms of fertilizer or irrigation applications, it'll consider that and inform your future recommendations. It's all anonymous, right? So none of the information you're putting in is available to anyone but you. Michael and others have done quite a few field trials at this point, testing the use of this tool compared to standard practices by specific growers, and the percent savings are super impressive, right? So on the nitrogen and the water side, saving anywhere from 20 to 40% has been observed, and so that really helps helps just demonstrate that by considering basic the soil properties, climate, etc., and using that information to make an informed decision, you can actually save on your inputs. And they saw no difference in yield, except in some cases yield actually went up. And so you're not necessarily incurring the risk of taking a hit on production by trying to use these sorts of tools. And UCCE staff are available throughout the state that have training and are available there as a technical resource for folks that are interested in learning this tool. There is an upcoming training in April out in Salinas with education credits available for that training. And I'll volunteer Jazz. I've talked with Jazz Breet, and she and others down in Kern are most certainly interested in understanding this tool and evaluating it in carrot production in Kern. And so over time, they will also become a very useful local resource for those growing in Kern about how to implement this tool. And then last but not least, through the SSJV MPEP, we have been awarded a grant from the CDFA through their Water Efficiency Technical Assistance Program. And so this grant is all about carrying out integrated water and nitrogen management trainings. And so getting into much more detail than I was able to today within a half an hour. Again, there will be continuing education credits available. We're looking for growers, advisors, all levels of the grower operation, consultants. Everyone is welcome. They're free. We're basically setting them up at 3 different locations within the SSJV. And so the carrot growers may be interested in the Bakersfield option. And we'll have a 3-part curriculum. And so the first part is kind of what I talked about today, but just trying to set the regulatory stage, understand the connection between management and impacts to water quality, and why nitrogen and water management is important from an environmental standpoint. And then we'll really get in the weeds on, specific irrigation and nutrient management practices for specific crop types, right? And so we'll have Jaspreet and others, take the time to talk specifically about carrots. And so you won't have to, unless you're interested, sit in the citrus section or the grape session. You can just attend the carrot session if that's your prerogative. And then we'll ultimately offer some actual tutorials of using crop manage. And so just additional opportunities to get trained up on that tool and understand how it works. And so, realizing that these water quality objectives are difficult and cumbersome, we're trying to, work with our partners at UC and amplify their knowledge and expertise and make sure that growers and advisors have the resources they need to actually achieve these water quality goals over time. And so I think that's all I really had for you today. I know that's a lot, but I'd be remiss if I didn't say all of it. So thanks for bearing with me.
Thank you, Ken. And we are already over time. So with that, I would like to say thank you to our speakers for their wonderful presentations. And thank you all for joining us today.